Hello and welcome to my channel, Vice-Rhino here. The following is a compilation of my Evidence for Evolution playlist where I took all 12 episodes and combined them into one long video. I'm going to leave a card to the original playlist, both because some people prefer to watch that way, and because starting in the geology episode, I began numbering my sources to make it easier to figure out what sources I used for what information. And rather than running out of characters in my video description here trying to copy all those sources over, it'll be easier for both me and you if I leave it as is, and if you want to know the source for a particular bit of information, head over to that specific video in the playlist and look for it in the description there. Also, there were two minor corrections found in pinned comments in the original videos. In the Vestigial Structures video, after making it, I learned that with regards to the appendix, there is evidence that the appendix helped our evolutionary ancestors in the digestion of plant matter, and as it lost that function, it gained its immune function, possibly performing both functions in tandem for a bit. And in the dating methods video, there was a minor correction to my explanation as to why radiometric decay occurs, which is too complicated to explain in a brief intro, but that's episode 5 in the playlist if you're curious. So, on to the videos. Hello and welcome to my channel, Vice-Rhino here. Today I'm starting a new series, and this is going to be a little bit different from my normal material. I have frequently made reference to the plethora of evidence that is out there regarding evolution, and how we know that evolution is a fact that is then explained by the theory of evolution. So in this series, I will do a deeper dive than I normally would into the different lines of evidence that support the theory of evolution. On to the video! I thought I'd start things off with some of the basics. So today's video is on homology and analogy. I'm sure most of my audience will have memories of high school biology class and the classic diagram of the forelimbs of the tetrapods presented as a prime example of homology. And that really is a prime example, so that's going to be our starting point. There is more to homology than just a loose similarity between different animals. It's about the basic structure. When comparing the bones of different tetrapods' forelimbs, we always see one single arm bone going from the shoulder or the shoulder equivalent down to the elbow. At the elbow, it connects to a pair of bones that go down the forearm to the wrist. In the wrist, there is a close grouping of distinct carpal bones, which then connect to the metacarpals and finally to the phalanges. This isn't just us looking at bones that have similar shapes and giving them the same name. It is the fact that the ones with the similar shapes are always connected to each other in the exact same order every time. There are no tetrapods with a radius and ulna combination on the upper arm and a single humerus-style bone below it. It is always shoulder, humerus, radius and ulna, then the carpals. They come in many different shapes and sizes, but always in the exact same order. This process of specific bone connections is most stark at the limbs, but it can be traced throughout the whole body. And what's more, the closer we are related to another organism, the more similar the bone structure will be. Because changes do happen, additions and subtractions do occur, but the overall plan is the same, and the order of the connections is the same. For instance, all vertebrates have tails. Every single one, including you. Your tail is just a stumpy little bone called the coccyx, though. Nothing to get excited about. Though it does start out as a bona fide tail in the uterus before the vertebrae fuse together into the coccyx. So unless you have a rare atavism, it stays under the skin. Do you know which other vertebrates have a stumpy little coccyx? The ones that seem to be our closest relatives when looking at other lines of evidence, like genetics. Specifically, the great apes. None of them have external tails, but they all have stubby little internal tails. Go a little further back, and you'll find that all mammals have the same 3 bones in their inner ears that if you trace them through embryonic development, they originate in a structure that forms the jaw bones in lizards. So it's not just that structures are similar that cause us to use homology as evidence for evolution. It is the fact that you can group animals by their homologous structures, and these groupings show a nested hierarchy of traits that matches closely with similar groupings that were arrived at through different methods, which I will cover later. And these nested hierarchies don't just group organisms that we see today, it fits in perfectly with the fossil record. The 3 middle ear bones that only exist in mammals also only exist in mammalian fossils until you go back in time far enough you reach lizard-like creatures that have mammalian characteristics, among them the 3 ear bones. Now these particular critters existed about 260 million years ago. If we go back another 40 million years, you get our common ancestor with birds. Birds have a very different system in place for hearing, but there are some similarities that are homologous, and this is where things get a little bit complicated, because if you remember, I had mentioned that we don't just look at things that are superficially similar, give them the same name, and then declare them homologous. But at this point, we run into the snag of having the same names for structures that are not homologous. The development of the tympanic membrane, for instance, didn't happen until after the lines that led to birds and mammals have separated from each other. So these are two structures that have the same function and look similar with the same name, but they are not declared homologous, because we are not just looking to show relatedness between every organism, we're looking to find out the actual degree of relatedness between organisms. Pretending that things are homologous when they are not is not helpful in this endeavor. And that brings us to analogous traits. We can also tell when two features are analogous rather than homologous, meaning that they serve the same function or have similar appearance, but do not share ancestry. Looking at the comparison of the mammalian and avian auditory systems, there are some aspects of them that are homologous, but others that are analogous. But the auditory system gets rather complicated, so I'll bring it back to the forelimbs, specifically the wings. Wings are a mixture of homology and analogy, as most things actually are when you do a deep dive on their evolutionary history. Looking at specifically tetrapod wings and ignoring the insect wings for now, they have the same bone structure all connected in the same order, but flight developed after the lineages that led to our modern flying animals diverged from each other. Now, how do we know this? Because the wings themselves are not homologous, even though the bone structures are. They accomplish the same purpose using the same bone structure, but in drastically different ways. The bat has a hand with elongated fingers, which then stretch out a membrane to form the wing. A bird has feathers extending from the back of the arm. Going back further, you can see pterosaur wings, which stretch a membrane from an elongated pinky finger all the way down to its ankles. Same bone structure, but different solutions for how flight would develop. This is also known as convergent evolution. Convergent evolution happens when the selection pressures of an environment strongly favor a particular trait, so that trait will develop more than once. Flight is useful, so it evolved at least 4 times: bats, birds, pterosaurs, and insects. Insects don't have bones in their wings, which you probably already knew, and since they don't have any internal skeleton to speak of, they don't fit into the homology described by the skeleton. Now, going on to another trait, sight. Sight is useful, so it evolved several times. There are possibly more than 50 independent lines of evolution that led to various types of eyes. And even this depends on where we draw the line for divergence. It is possible that some basic form of photosensitivity developed very early on, and that all eyes share this basic homology, but built on it in different ways. So if we are just looking at the ability to sense photons, that trait could be said to be homologous between most, if not all, animals that have any sort of light sensitivity today. But then when we look at the details, we can see fundamental differences in how they go about sensing and processing light. Vertebrate eyes are very different from cephalopod eyes, which are very different from other mollusk eyes, which are very different from insect eyes, etc. So we know that vertebrates and cephalopods did most of their eye development independently from each other. We can tell this by examining the various structures. In particular, the photoreceptor cells in both lineages are facing opposite directions, with vertebrate photoreceptors facing the back of the eye, and cephalopod photoreceptors facing the lens. This is a development that would have had to happen very early on, and as evolution has no final goals, there wouldn't necessarily have been a disadvantage to having the photoreceptors facing backward early on, and there was no plan to deal with the problems that would crop up as a result of this, let's call it a mistake, when more complex eyes developed. So now all vertebrates have a blind spot where the optic nerve that carries the information from the photoreceptors to the brain has to pass through the retina in order to get that information to the brain for processing. Though if you want to get technical, our eyes are basically physical extensions of our brain that are exposed on our face. Bit of a weird thought, but it is true. Also worth noting here is the nautilus eye. This is another example of evolutionary biologists not just finding similarities and declaring them homologous for the sake of making everything fit their predetermined notions of evolution, but rather investigating and changing their conclusions based on new information. There are 2 living clades within cephalopoda, the coleoids and the nautiloids, with the nautiloids being represented by one single genus, the nautilus, and the coleoids being all the rest of them, the squids, cuttlefish, octopuses, etc. The nautiloids have a very different eye from the coleoids. Their eyes are basically like a pinhole camera with no lens or cornea. The nautilus is also what's known as a living fossil, that is, a currently extant creature that is very similar, at least superficially, to its ancestors' form millions of years ago. The logical conclusion of these differences is that the coleoid cephalopods diverged from the nautiloids before the development of the lens and the cornea, which allowed them to develop more sophisticated eyes and left the poor nautiloids in the dust. But fairly recent research has determined that this is not likely the case. A genetic examination and comparison of nautilus and pygmy squid embryos has determined that the nautilus has the necessary genes for producing the cornea and lens, but these genes are just not expressed in the same way as for the coleoids. This suggests that their ancestors did have eyes similar to the coleoid cephalopods, but at some point in the past lost the ability to grow a lens or cornea, providing a good example of divergent evolution where one feature changes in different ways over time in several different lineages. Now, I have drastically oversimplified the process by which we determine whether or not different structures are homologous. There's genetic analysis, statistical analysis, protein analysis, there's debate within the scientific community, and eventually they do come to a consensus, usually. I've used examples that are so obvious that it's easy for someone with no training to see, but as I've been researching the matter, I keep coming across papers and articles that go into great detail on which individual proteins are homologous between which organisms, and how the variants of these individual proteins developed independently in other organisms and whatnot. In looking into the eye, the protein crystallin, which is the transparent protein that makes up the lens of the eye, I found that it has at least 89 variants that are found in humans alone, and they vary in length from being as short as 26 amino acids to 34,350. And within the human body, some of these crystallins are active enzymes, while others are inactive, but they have been determined to be somewhat homologous to other active enzymes. So within our own body, we have structures that are homologous to other structures in our body, which itself is a bit of a mind-bender, but it makes sense in light of evolution. After all, the idea is that all of our structures evolved from something that existed already in the past, but in the case of something like a protein, there isn't necessarily a reason to stop producing the previous version once a new version develops, so we can see both versions being actively produced by the body now. To conclude, homology and analogy are not determined arbitrarily based on what scientists determined in advance must have been the case. Different structures are carefully studied, data is collected and then analyzed, and then based on this observable data, structures are classified as homologous or analogous. And by using this data, organisms can be placed in categories based on how closely homologous their structures are, and these categories match up very closely with other methods for grouping organisms together, and it shows degrees of relatedness. Homology alone is excellent evidence for evolution, but it is not alone. Join me next week as we look at the evidence for evolution found in embryology. For early access to my Friday videos, consider becoming a patron. You can also follow me on social media or donate through PayPal. Links to all of that are in the description. See you next time. Hello and welcome to my channel. Vice Rhino here. Today we're continuing on with the Evidence for Evolution series. This time we're going to take a look at embryology. But before we begin, just a clarification. This series is supposed to be educational. I will not be making reference to creationist arguments throughout this series, as it is not a response to creationism. If you want to see me directly deal with creationist arguments, that happens on Fridays, Thursdays for patrons. Part of the purpose of this series is for me to use as a reference when I am dealing with creationism, so when I say that there is evidence contained in embryology for evolution, rather than hunt down all the papers again, I could just point to this video. It is also meant to be approachable to the people that do hold the creationist viewpoint. It is somewhere they can come to see why people think evolution is true without having to tolerate a direct attack on their belief system. I am not actively trying to change anyone's position with this series, it's just laying out the evidence that we have in a way that is hopefully easy to understand. So let's go! Embryology is the study of the prenatal development of an organism, and can encompass everything from the production of gametes to a fully fledged, ready-to-be-born or hatched fetus. The evidence for evolution found in embryology is often along the same lines as the evidence found in homology, to the point that while making my homology video, I found it incredibly difficult not to use embryological examples, as I knew that embryology would be a different topic by itself. In fact, one of the ways we can tell if a structure is homologous is to trace its embryological development. A truly homologous structure will originate in the same place of the embryo, while an analogous one may not. Embryology is an interesting field because we can almost see the actual evolutionary development of an organism in the stages of the embryo. Not a perfect reenactment of evolutionary history like Ernst Haeckel proposed with recapitulation theory, but it holds true that the more closely related two organisms are, the more similar their embryological development will be, regardless of how similar their adult forms are. And as with homology, there is more to the similarities than a simple superficial resemblance. So let's start off with the gill slits. This is one of the easiest examples, as it is quite obvious just by looking at the different vertebrate embryos that they all have the same structure on the neck area that look remarkably like the structures that turn into the gill arches of fish. But the similarities go deeper than just a superficial resemblance. For instance, the structures originate from neural crest cells in the embryo. Neural crest cells are cells that form in the embryo just after the neural tube forms. In the embryo at this stage, there are essentially 3 layers of cells, the germ layers, with the top layer being the ectoderm, the middle layer being the mesoderm, and the bottom layer being the endoderm. The neural tube is formed when the ectoderm basically folds in on itself, and the neural crest cells are the ones that are on top of this fold but just under the surface. I tell you this because we can trace individual cell histories and see exactly which area each structure originates from in the embryo. This is important because most bones trace their origin to the mesoderm, but the jaw bones that come from the gill slits trace their origin to the neural crest cells. And the gill arches in jawless fish also trace their origin to the neural crest cells. The odds of 2 unrelated features sharing an origin path throughout embryological development are extremely low, especially when we consider that the non-fish version turns into a structure, namely bone, that normally has its origins elsewhere. There's also the fact that the musculature of the vertebrate jaw appears to be homologous to the musculature of basal gills. And then there's the upper portion of the second arch, which turns into a structure called the hyomandibular bone in jawed fish, which acts as a support for the skull. A similar structure is found in modern reptiles, which not only helps to support the skull, but is pretty good at conducting sounds and has become part of the reptilian auditory system. And it originates from the same place on the second arch. The stirrup bones in the mammalian middle ear also originate from the upper portion of the second arch, but it no longer does any work in supporting the skull. As terrestrial vertebrates spent more time out of water, their cranium became more firmly attached to the rest of the skull, and thus no longer needed the support of the hyomandibular bone. But by the time this had happened, it had already started acting as a sound transducer, so its secondary function then became its primary function over evolutionary time, and it became the stirrup bone. And what's more, we find evidence that the ancestors of mammals diverged from the ancestors of birds and modern reptiles before the jaws developed the articulation that we see today. We can tell because the quadrate and articular bone in amphibians, reptiles, and birds all develop from the same part of the cartilage of the first embryonic arch. These are the bones that are responsible for the articulation of their jaws. But in mammals, the articulation happens in the dentary and squamosal bones, which develop from a slightly different section of the embryonic arches. This allows the bony elements that become part of the jaw in reptiles, amphibians, and birds to develop into the bones of the middle ear, with the quadrate becoming the incus bone and the articular bone becoming the malleus. By tracing the developments of these bones through the embryo, we can see that the posterior lower jaw bones of reptiles, the gill arches of jawless fish, and the middle ear bones of mammals are all homologous in that they share their origin in the embryonic gill arches. And we can tell how closely related different animals are by tracing these structures' development through the embryonic stages. So now let's take a look at snakes. Snakes, despite not having any limbs, are tetrapods, which literally means four-footed. So why would we call them tetrapods? Well, it's because we know they are descended from tetrapods. How do we know? Well, one of the ways we know is that the embryos of some types of snake start to develop legs in the first 24 hours, complete with rudimentary leg bones like the femur, tibia, and fibula. But they don't make it far enough into differentiation to actually become cartilage, much less bone, so they end up degenerating. There are even more leftovers of the evolutionary process found in embryology. One of my favorite examples is the blowhole of the dolphin. As an early fetus, the dolphin has its nostrils on the front of its face, matching up with all the rest of the mammals. But as the fetus develops, the nostrils slowly migrate back to the top of the head. And these dolphins, with their nostrils, still have gill slits. And the gill slits in these dolphins still develop into ear bones, and which arch develops into which bone matches up with what would be expected of mammals. But there are several adaptations that are specific to hearing in the water, like the fat pad. And the dolphins still have evidence of hind limbs in their embryonic stages, despite not having any sign of external hind limbs. Unlike the snake limbs, though, the dolphin's hind limbs turn into a rudimentary pelvis, which still serves the function of anchoring some of the muscles associated with mating. Evolution has a way of taking old, defunct bits and finding new purposes for them. And that makes sense, because evolution does not have a plan for the future. It just works, through natural selection, with the variation in what is currently available. We can also bring this back to the tetrapod forelimb. Remember how we all have a humerus connected to the radius and ulna, connected to the carpals, then to the metacarpals, and then the phalanges? Well, as it turns out, one of the features that makes a tetrapod is that it has 5 digits. But looking at some of the tetrapods, we don't see 5 digits. I mean, we even categorize some of them by how many toes they have. Being an odd-toed ungulate myself, I am well aware of this fact. But when we look at the embryo, all tetrapod embryos go through a stage where they develop limbs with 5 digits, with the exception of the snakes whose limbs are reabsorbed before becoming prominent enough to count fingers. These digits will sometimes fuse together into slightly different structures, but they always start with 5. This is also another way we can tell that the tetrapod forelimbs are homologous rather than just having a superficially similar appearance. Remember how we can trace the formation of the structures in the body all the way back through embryonic development? All the bones in the forelimb share the developmental path. They all originate within the same areas of the embryo. There is no reason at all that all tetrapods should start with 5 digits. There is no inherent advantage or disadvantage to it. There's no reason it should be 5 rather than 6, or 4, or 12. But it is 5, because evolution has no plan. When the first tetrapods evolved, for whatever reason, the ones with 5 digits on their limbs were the ones that were successful. So now all tetrapods start with 5 digits, even if 1 would have been better, like horses, or 3, like in birds and rhinos. Evolution works with what it has been given by past evolution. The successful tetrapod ancestor had 5 digits, so its descendants all have 5 digits. In environments where fewer digits were advantageous, it is easier to select for the fusing of digits, which is a fairly common mutation, rather than going back and redesigning from scratch. How would evolution redesign from scratch anyway? It would have to be a massive series of serendipitous mutations all happening within extremely short periods of time, like we're talking 1 or 2 generations, which I suppose is technically possible, but it is incredibly unlikely. What is likely is that fairly minor changes in existing structures could be useful in a new environment, and the fusing together of the digits is a common enough mutation that it is completely unsurprising that in some environments it was useful enough to stick around. Heading back into the ocean, we have baleen whales. Baleen whales have evolved from ancestral whales that had teeth. There is a decent, if a bit spotty, record for the evolution of the baleen. It looks as though the whales lost their teeth first, becoming suction feeders, much like the modern beaked whale, which has neither teeth nor baleen, with the baleen being a later development. We can tell through examining them and through the fossil record that baleen and teeth are not homologous. That is, baleen are not simply specialized teeth. They are an unrelated structure. But if we look at the embryos of modern baleen whales, before the baleen even develop, teeth are formed beneath their gums and are then reabsorbed. There is no reason for these teeth to develop. They never even make it out of the gums. They don't turn into anything else. They aren't useful. They just form and then disappear. The only reason baleen whale embryos should have teeth is if they evolved from a toothed whale that lost the ability to make teeth. It is possible that the reason they haven't completely lost their teeth yet is because the baleen might develop through a similar genetic pathway as the teeth. This makes sense because the teeth continue to grow well into mid-gestation, which seems like a really big resource drain that would be selected against during the millions of years of evolution since the baleens first developed. But if the baleen production uses proteins that are coded for by the genes that make the teeth, then this perfectly explains why the beginnings of teeth continue to linger despite their apparent uselessness. We can also look at the muscle structure of the human embryo for evidence. Human embryos have more muscles than adult humans have. Some of the muscles disappear completely, similar to the teeth of the baleen whales. Others fuse into different muscles. But we can look at these muscles and see homology between human embryos and other species. For instance, human embryos have a muscle in their forearm, the epitrochlear anconeus, which can also be found in adult chimps. In most cases, this muscle disappears before birth, but some people can retain this muscle into adulthood. The same is true of the dorsometacarpalis muscle, which is found in modern lizards, but not found in mammals, except for fairly rare atavistic cases. An atavism is when a vestigial form has been lost in a species but makes a reappearance through a mutation. These muscles are still found in human embryos, developing in the exact same way as they do in lizard embryos. In embryology, embryo formation tends to be conserved. Structures can be found in embryos that are completely absent in adults. Sometimes these structures diversify in different ways in different organisms, such as the gill slits turning into gill arches, jaw bones, and ear bones. Also the tetrapod limbs starting with 5 digits but diverging as the embryo grows into single-toed hooves, three-toed hooves, three-fingered birds, etc. Other structures do their best to disappear completely, but still stick around despite being a pain in the butt, literally in the case of the coccyx. There is no reason for embryos to go through the motion of forming body parts that don't end up developing unless they are leftover remnants from an evolutionary ancestor. Horses don't need 5 digits in the embryonic stage, they just need the one that they end up with. Baleen whales don't need embryonic teeth, but they have them for a while. And sometimes it's a good thing that these structures stick around. Had the hyomandibular bone just gone away when we stopped needing it as a support for the skull, then we wouldn't have the ability to hear like we do today. It's possible, probable even, that it would have evolved in a different way, as hearing is a very useful feature. After all, hearing has evolved independently at least a couple of times. But evolution took a bone that we didn't need anymore but happened to be good at transmitting sound, and adapted it into a bone that specializes in hearing. Of course, I am speaking anthropomorphically here, projecting intent on an unintentional process, but that just makes it easier to wrap our heads around. So that's it for today. Join me next week as we take a look at vestigial structures. Hello and welcome to my channel, Vicedrino here. Today we're continuing the Evidence for Evolution series with the topic of vestigial structures, and I promise this is not just an excuse for me to say coccyx a bunch of times, so let's go! First, it is helpful to define what a vestigial structure is. Most definitions state that it's a structure that has no apparent function and appear to be residual parts from a past ancestor. But it is important to note here that we do not need to assume common descent for something to be vestigial. We can just compare the vestigial structure with a currently functioning version of the structure in a different species. For instance, the wings of flightless birds. We don't need to assume that flightless birds had a common ancestor with birds that can fly. We can see that the structure of the flightless wings is homologous to the structure of the functioning wings, but they can't fly, so it's vestigial. Cave-dwelling salamanders have eyes that start to develop into the early stages of the salamander's life, but then regress and get covered over with flaps of skin. Whales and snakes have pelvises, but no hind legs, and things like that. Also important is the fact that a structure does not have to be completely devoid of function in order to be considered vestigial, but the function that it currently has does have to be different from the function that it is a remnant of. After all, can you really call a wing a vestigial wing if the animal still uses it to fly? So even though there is evidence that cetaceans use their vestigial pelvises as anchor points for muscles that are important to mating, the fact that they no longer use their pelvises to connect to legs makes their pelvises vestigial, since that's the main function of pelvises in other animals that have them. So it is a definitive fact that vestigial structures do exist. Now, why are they evidence for evolution? Isn't evolution about building new things rather than dismantling old things? Well, yes and no. Evolution is an undirected process. There is no end goal. A mutation doesn't happen with the goal of eventually building an eye. But if a skin cell mutates in a way that makes it sensitive to light, and this sensitivity helps the organism survive long enough to reproduce, then that mutation will survive. Actually, I should clarify, the mutation would have to be in the germline, not the skin cell itself. A germline mutation is a mutation that occurs within a germ cell, which are the cells that develop into sperm and eggs. The mutations that are heritable happen in the germ cells. So for this light-sensitive cell to contribute to evolution, there would be a mutation in the germ cells that cause some of the skin cells to have light sensitivity once they develop. Evolution can then take this light sensitivity to light and build on it through more germline mutations. But then what happens to this hypothetical eye when an organism's environment changes? So let's say a salamander somehow ends up living in an underground water system with no light. Well, in that scenario, having the ability to detect light will be pretty much useless, so there is no more evolutionary pressure to produce and enhance the salamander's already existing eyes. But there's also not necessarily pressure to completely eliminate it either. But with no pressure to either improve or eliminate a feature, the tendency is for features to atrophy over time. Without a selection pressure to improve on an existing feature, the random germline changes that will have an effect on the feature itself will tend to degrade it. In fact, there could even be a slight benefit to degrading a useless feature. It takes a lot of resources to build a fully functioning eye. Those resources could be put to use elsewhere in the body if they weren't so busy building eyes. So in this instance, there will be a very slight selection pressure toward the elimination of the eye, which then frees up these resources for other uses. In fact, it's not hard to imagine in these salamanders' case why there might have been a selection pressure in favor of a bit of skin covering their eyes. After all, eyes are tender and vulnerable. If you're not going to use them for seeing, then it's just a spot on your face where it's easy to get hurt. So with an evolutionary process, one would expect that, over long scales of time, you would see structures that used to perform a function in the past that no longer work for that function. And this is what we see. But more than that, because evolution can only work on structures that already exist, you would expect to see structures showing signs of changing what their primary function is. So, for instance, the pelvis in the whale. We now know that this serves as an anchor point for muscles that are important for mating, but how do we know it was really a pelvis and not just a similar-looking structure? Because we're able to trace its embryological development, and it shares the development path of other mammalian pelvises. So we have vestigial structures that become atrophied by a combination of random mutations and a lack of selection pressures. Does this mean that mutations can bring these structures back? Sometimes. When a vestigial structure mutates in a way that causes it to become more fully developed than it normally is, that's called an atavism. So for instance, as an embryo, all humans had a tail. It started off fairly small, but gradually grew until its length peaked around Carnegie stage 16, Carnegie stage is just being a measurement of the development of the embryo. After Carnegie stage 16, the tail starts shrinking, until finally turning into the fused vertebrae that are our coccyx. But sometimes, because of mutations, the tail continues to develop into an almost full external tail. Depending on the exact cause of the tail, some people can actually control it and move it. Now let's look at our eyes. In the corner of your eye, you have a small flap of skin that is all that remains of our ancestors' nictitating membrane. The nictitating membrane is the third eyelid that many animals use to protect their eyes and keep them moist, while being at least partially translucent so they can still see through it. It's present in birds, reptiles, and most mammals, but is very rare in apes. We can see the vestiges of the nictitating membrane in our Plica semilunaris, but we don't get a full third eyelid that we can sweep across our eyes. And it is again worth pointing out that this has been determined through embryological study. The Plica semilunaris is the result of the embryonic version of the nictitating membrane, and as with so many vestigial features, it develops further in the embryo than it needs to, and then regresses and is reabsorbed in later stages of development. So as an embryo, we develop a nictitating membrane that covers the full eye, but then shrinks, or rather doesn't grow as the eye grows, until it's just a bit of skin in the corner of the eye. But sometimes, an incredibly rare mutation will cause the nictitating membrane to survive embryonic development and persist into childhood, such as in this case here with a 9-year-old girl. This was not a fully functioning nictitating membrane, and we wouldn't expect it to be. We would not have lost the nictitating membrane in one single large mutation, so why would we expect it to come back in one big leap? But this girl had a nictitating membrane that was attached to the Plica semilunaris in exactly the way one would expect of a bona fide nictitating membrane. And in a standalone video on vestigial structures, I would be remiss if I didn't mention the appendix. It has been thought since Darwin's time that the appendix is a vestigial structure, a leftover from our herbivorous ancestors that aided in the digestion of plants. However, this has turned out to not be the case. This myth has persisted largely because an appendectomy is one of the most common surgical procedures in the developed world, and for most people, there are no long-term side effects of having your appendix out at all. But it has been suggested from as early as 1900 that the appendix has a function as part of the immune system. Important to note here that this hypothesis was made on the grounds of phylogenetic study, with phylogenetics being another line of evidence for evolution that I will cover in a later video. For now, let me just say that using evolution as an assumption, the prediction was made that the appendix serves a function as part of the immune system. It wasn't known what the function would be, and many a joke has been made over the years that the primary function of the appendix is to pad a surgeon's pockets, but in 2007, it was identified as being essentially a reservoir for a person's healthy gut bacteria to repopulate the intestines during recovery from an infection that is cleared out by a rapid flushing of fecal matter from the colon, known more commonly as diarrhea. It took so long to discover this function of the appendix for two reasons. Firstly, it wasn't until 2003 that it was discovered how important the colonies of symbiotic bacteria living in our digestive tracts are to our health and immune system. So the fact that the appendix served as a safe house for this bacteria wasn't something anyone would have even thought to look into, for the simple reason that they didn't know how important these bacteria are. And secondly, because the apparent lack of long-term health consequences from having the appendix removed supported the idea that it didn't perform any sort of function. But this is where it gets kinda neat. We are actually living through a period of time where the appendix is likely to come out on the other end as a vestigial structure, because the reason you can live a perfectly healthy life without one is because of modern medicine and hygiene. Historically speaking, diarrheal diseases have always been one of the top causes of human death, so the existence of a feature that evolved to help protect us from these illnesses is expected, and of course it doesn't work perfectly. Its role is essentially to help with recovery. If the disease is caused by poor sanitation, and the sanitation doesn't change, the person won't get to a point where repopulating the gut bacteria will matter. But in the instances where they do successfully eliminate the disease, the appendix is very helpful for a safe recovery. So in developed countries with access to modern medicine and clean drinking water, I guess that means not Flint, Michigan, sorry guys, the appendix is essentially useless, making it an excellent example of an organ that, while not technically vestigial, is on the road to vestigiality. It is entirely conceivable that since modern medicine and hygiene have rendered the presence of an appendix unnecessary, and that while appendicitis is usually not deadly, it does still kill a few hundred people every year, that there is actually a slight selection pressure toward the elimination of the appendix. So it is possible that in a few hundred to a few thousand years, the appendix will be greatly diminished from what it is now, or will have developed some other function than the one it serves today. I could go on and on about all the different vestigial structures that are out there, from our ear muscles that actively try to turn our ears towards sound despite them being too weak to actually accomplish this task, even in people that can still wiggle their ears, to male nipples, to lizards who exist in an all-female population but still go through the motions of sex with each other before reproducing through parthenogenesis, which is the laying of an unfertilized egg that still develops, to dandelions still having sex organs necessary for sexual fertilization despite reproducing entirely through asexual means, to goosebumps. All of these are fascinating examples of vestigiality, and when tied in with homology and embryology, we can tell in most cases that these vestigial features are homologous with fully functioning counterparts in other species. This only makes sense in light of evolution, that as a species adapts to a changing environment, it ceases to need something that was helpful for their ancestors, and since there was a lack of selection pressures, mutations over time cause that feature to atrophy, or in some cases, develop a new use for which there is a selection pressure. Thanks for watching. If you liked this video, don't forget to subscribe. There are new additions to this series every Monday at 9 AM Eastern, and I do direct response videos to creationists usually every Friday at noon Eastern. Special thanks to my patrons who are the super lube that keeps this machine greased up. And if you want to be my lube, you can support the show at patreon.com/ViceRhino, or just follow the link in the description. Links to my social media pages can also be found in the description. See you next time. Hello and welcome to my channel. ViceRhino here. Today we are continuing on with the Evidence for Evolution series, but this time we're going to veer away slightly from the topic of evolution itself, as we need to have a basic understanding of geology in order to fully appreciate just how much evidence for evolution there is in the fossil record. So today I'm going to be covering some aspects of geology. I have also been asked to make it easier to tell which source I am using at which times in the video, so I have now numbered my sources and will display which number I'm referring to on screen. So let's go! Geology is a big topic. I couldn't possibly cover the entire range of subjects that are encompassed by the term geology in one single video. But as geology is not itself evidence for evolution, I will do my best to cover the relevant aspects here. The relevant aspects being how we can tell past environments from geology, and how we can determine ages. I will spend this video discussing the different environments that we see in the different strata. Strata, put simply, are the horizontal layers that can be found in sedimentary rock. They can be paper thin or several meters thick, depending on how they are formed. But that's the general idea. Geologists can tell, by looking at the different characteristics of the strata, what types of environments they were deposited in. So, for instance, tillite tends to lack internal strata, be poorly sorted, with grains like pebbles and gravel being completely surrounded by the finer grains, will be massive, and will contain few fossils. Tillite is the result of glacial ice eroding rock as it moves across the continent. Picture it like pushing sand on a beach. The sand gets moved from where it was and pushed forward as you move. It doesn't discriminate based on grain size. A glacier will push a boulder just as well as fine grains of sand, so it ends up as a poorly sorted sediment. Because of the nature of the deposition and how slowly it forms, it is unlikely, though not impossible, that glacial deposits will bury an organism's remains fast enough for fossilization, and so not many fossils are expected. Tillite is actually one of the lines of evidence for continental drift. Because it is so indiscriminate in its makeup, if you can find two beds of tillite on opposite sides of the ocean that have closely matching characteristics, you can be fairly certain that it is actually one bed that has been split over geologic time. And we do find beds like that. And matching the tillite is more than just looking at the characteristics of the tillite itself. It is also looking at what kind of strata are surrounding it and matching up the sequence of strata. And we do know that it is formed by glaciers because we can see active glaciers depositing sediment in a similar fashion today. This is one of the keys to a lot of these geologic environments. We know how different environments deposit sediment differently because we can find examples of those types of deposition happening today. But let's back up a bit. Tillite is a sedimentary rock. Sedimentary rocks are rocks that are composed of other rocks that have been eroded in one way or another and then reformed into its own rock. So sedimentary rocks are rocks like sandstone, which are made up of grains of sand, or limestone, which are usually made up of skeletal remains of marine organisms like coral and mollusks, which are then subject to enough pressure to cement the sediments into rock in a process called lithification. Metamorphic rock is rock that has undergone metamorphism, which are changes to the mineral structure and makeup of solid rock, usually by high temperatures and pressures. Basically, it's a rock whose physical properties are changed without quite melting it all the way into magma. And that's where igneous rocks come in. Igneous rock is rock which has been completely melted into magma, or never solidified in the first place, and either cools slowly under the surface, working its way toward the surface over thousands to millions of years to form intrusive igneous rocks, or exiting by way of volcanoes and fissures to cool quickly on the surface. We can also tell when sediment is deposited by a river, known as fluvial deposition. Fluvial deposition can usually be split into two categories, the riverbed itself and the alluvial fan. An alluvial fan is similar to a river delta with one main important distinction. Deltas are when the mouth of a river opens into a standing body of water, while alluvial fans are when a river or stream spreads into a receiving basin that is not already occupied by water. The fan itself is conical in shape and is usually made up of conglomerate or arkose rocks, and the sediment in a fan is typically gravel-sized and shows a decrease in grain size as you move further away from the mouth of the fan. It's even possible to determine whether the sediment in the fan originated as part of a landslide or if it was just a normal stream flow deposition. There are also instances where geologists can tell that the fan is the result of a catastrophic sudden discharge of water resulting in a sheet flood. And because fans tend to be high-energy oxidizing environments, there are not many fossils to speak of. Some examples of ancient alluvial fans include the Hornellen Basin of Norway, the Gaspé Peninsula in Canada, the Mount Toby conglomerate in Massachusetts, and the Todos Santos Formation in New Mexico. And since we started with alluvial fans by comparing them to river deltas, let's just take a look at deltas next. Deltas happen where rivers meet larger bodies of water. Rivers carry sediment with them as they flow, and when they flow into a larger body of water, they slow down. This slowing causes them to drop the sediment that they had been carrying, creating a pile of sediment on the floor of the larger body of water. Eventually, the sediment piles up to a point where it is above the surface of the water, and becomes dry land with the river forking around it. Then the two new forks begin the process again, and over time, as the river keeps forking around, the deltas become larger and more complicated. In deltas, we usually find marine and non-marine mudstone, siltstone, sandstone, and even coal. There will also be ripple marks in the rock as the result of the waves and the current action on the sediment. There are also different types of deltas which depend on the relative density of the river water when compared to the water that it's flowing into. When the river water's density is roughly equal to the density of the water it's flowing into, there's a rapid mixing of waters resulting in an abrupt deposition of the sediment load of the river, and this is most common at the mouths of coarse-grained rivers. If the river water is more dense than the water it's joining, which is common in flood events, then the river water forms a current that moves along the bottom and deposits its sediment on the more gentle slopes of the delta to form turbite deposits. And thirdly, and probably most common, is when the river water is less dense than the water it flows into, which is common where rivers meet oceans and seas, as salt water is more dense than fresh water. In these conditions, the river water forms a current on top of the salt water and can flow quite some distance away from the shore, carrying sediment the whole way. Eventually, positive ions in the seawater neutralize the negative charges on the clay particles, causing the aggregation of fine sediment into small lumps in a process known as flocculation. This tends to make for a larger delta with a very shallow dip of about 1 degree, as opposed to the 10 to 20 degree dips common in the other types of deltas. Deltas are one environment that is known as a transitional environment. That is, it is an environment that marks the boundary between two different environments, the two environments in this case usually being fluvial and shallow marine. Common fossils in delta formations include terrestrial plants, mollusks, and bioturbation, which is just geology talk for the sediment having been disturbed by an organism before it lithified, and that disturbance is captured in the fossil record, so footprints and the like. Some examples of ancient deltaic systems include the Wilcox Group of Texas, the Dunvegan Formation in Canada, the San Miguel Formation in Texas, and the Misoa Formation in Venezuela, which is one of the larger deltas extending about 250 kilometers from its source. Now let's talk about the Aeolian desert environments. Usually when we think of deserts, we think of large, hot areas covered in sand dunes. But in reality, a desert is simply defined by how much rainfall it gets per year. And by this measure, the Sahara is only the third largest desert in the world, with it being beaten out by Antarctica and the Arctic. So there are a myriad of sub-environments in the larger category of desert. Alluvial fans are actually one common sub-environment, but also there are intermittent streams, intermittent saline lakes, sand dune fields, sediment-covered interdune areas, and bare rocks. In desert Aeolian environments, for obvious reasons, we usually find sandstone, and the fossils that are found are usually terrestrial reptiles. But deserts aren't the only kind of Aeolian environment. Any sediment that is deposited by wind is Aeolian sediment, and one of the most common forms of aeolianite is the coastal limestone made up of the carbonate sediment that is the result of shallow marine organisms like the seashells of mollusks. In order for this type of aeolianite to form, there needs to be a warm climate, enough wind to form sediment from the beaches into dunes, and relatively little rainfall. Now that's relatively little rainfall, but it does need some rainfall to percolate through the sediment in order to facilitate lithification. Last up, for sedimentation environments, I want to talk about one particular aspect of marine sedimentation, the formation of chalk. Chalk is essentially an accumulation of a specific category of microscopic plankton, coccolithophores. Coccolithophores have a spherical skeleton referred to as a coccosphere, which is made up of discs of calcium carbonate. When the plankton dies, its skeleton sinks to the ocean floor. Over time, these skeletons would accumulate, and this is an incredibly slow process that requires relatively calm waters. Turbulent waters would have easily swept up the discarded coccospheres into other depositional environments which would have had other sediments that accumulate faster, and so would greatly outnumber the coccospheres. Now, in a stroke of serendipity, my son actually came home from school today after learning about geology with a vial of water in which had been placed gravel, sand, and chalk dust. After shaking the vial, the gravel sinks to the bottom almost immediately, with the sand settling in not too long after, but the vial has been left undisturbed for hours now, and the water is still blue with the chalk sediment floating in it. This is actually a good example of why you need a calm environment in order for the coccospheres to be able to sink to the bottom. With turbulent water, it would just get swept up in the water itself and would never quite settle. The deposition of chalk requires a delicate balance between how many coccolithophores are alive near the surface of the ocean, the rate at which they dissolve once they reach the bottom, and the rate of accumulation of terrigenous sediment, that is, sediment that is washed into the ocean from terrestrial environments. If there's too much carbon dioxide dissolved into the water, then the coccospheres will dissolve before they can become part of the calcareous ooze that would eventually lithify into the chalk beds. If there's too much terrestrial sediment, it will overpower the chalk. And the waters had to be relatively calm for extremely long periods of time. The calcareous ooze accumulates at a rate of about 1 to 6 centimeters per 1,000 years. We can even tell from looking at the chalk beds when the climate was more conducive to their formation. There is evidence of cyclic deposition, where there are periods of low energy and steady accumulation rates, alternating with interruptions of deposition by high energy and possibly shallower waters. We also know that the calcareous ooze that results in chalk can only form in water that is less than 4,500 meters deep, with the estimate for the waters being around 200 to 300 meters deep. The chalk beds of South England contain 3 subdivisions, dubbed the lower chalk, the middle chalk, and the upper chalk. The contacts between these layers are a gradual gradated change, which indicates that the environmental changes that led to these distinct layers were also gradual changes. And now that we know some of the basics of sedimentation, we can move on to stratigraphy. Stratigraphy is the study of the geologic strata, concerning the order and relative positions of the strata and how they relate to the geologic timescale. When looking at the rock layers, we can find strata that are laid down in the exact same order, and which share otherwise unique characteristics, but they're spread out across continents. An example are the Appalachian Mountains of North America, which have a very close continuity with the Caledonian Mountains of Scotland and Scandinavia. This was the example that led to the original proposal of continental drift by Frank Bursley Taylor. The study of strata and the matching of stratigraphic sequences in different locations have helped geologists piece together the history of the Earth, leading all the way back through the 7 major supercontinents. The geologists who study stratigraphy use known information about how different types of sediments are deposited in different environments to create maps of the Earth as it would have been during the different time periods represented in the strata. And I have only barely scratched the surface with regard to how detailed the study of sedimentation can get. Stratigraphy has, in the last half of the 20th century, matured into an incredibly accurate science, which is now used to predict the locations of petroleum and natural gas deposits. So in summary, we know an incredible amount of detail about where and how different types of rocks form, and how long these processes take. We then use this knowledge to map out the geology of the past, comparing the different rock layers to one another in different locations, and coordinating locations using the matching of sequences in the strata. Next week we'll be taking a look at the different dating methods that they can use to determine the ages of the rocks. Thanks for watching. If you're enjoying this series, remember to subscribe to be notified of new episodes, and consider becoming a patron at patreon.com/ViceRhino. Patrons are what make this series possible, and I mean that in the most literal possible sense. Patrons also get some nice perks, like early access to my Friday videos. This past week, my patrons got early access to one Apologia's videos as well as a nice little bonus. And I'd also like to give extra special thanks this week to Stephen Bauman, who is a geologist who is kind enough to let me bother him from time to time when I get stumped. So thank you very much, Steve. See you next time. Hello and welcome to my channel. ViceRhino here. Today we're at number 5 in the Evidence for Evolution series. My next video that will be providing direct evidence for evolution is going to be on the fossil record. So before I get into the fossil record, I want to give some background as to how we know some of the things that we know about the fossils. Last week I talked about depositional environments and how rocks are formed, which helps us understand how we know what type of environment the fossils were in when they were alive. This week, I'm going to be talking about dating fossils, which is an appropriate subject with Valentine's Day coming up. So how do we know how old a fossil is? We're going to start out with some of the different dating methods and finish up with relative dating and stratigraphy. So let's go! I'm going to start off today with a dating method that likely most of you have never heard of, electron spin resonance dating. This is only possible with insulating minerals. An insulating mineral has 2 energy states that an electron can occupy, the low energy valence band and the higher energy conduction band. When the mineral is formed or reset, all the electrons will be in the valence band. Radiation from elements in the mineral and from its surrounding material will sometimes knock an electron into the higher energy conduction band, which then leaves a positively charged hole in the valence band. Most of the electrons will, in short order, recombine with these holes and end up back in the valence band, but some of the electrons will end up trapped by defects in the crystal lattice of the mineral. These electrons can be directly measured with a process called electron spin resonance spectrometry. The measurements of these electrons essentially measures how much of a dose of radiation the sample has accumulated since being formed or reset. Measurements will be taken of both the material being dated and the rock surrounding the material in order to determine the dose rate, that is, the rate at which it was being exposed to radiation. Determining the age then becomes a simple matter of dividing the accumulated dose by the dose rate. The most complicated part of this dating method is determining the dose rate itself. In determining the dose rate, it is necessary to calculate the internal and external dose rates independently, with the internal dose rate usually being different due to a difference in the concentrations of the various radioactive elements and their ranges. And as part of the external dose rate, the effect of cosmic rays also has to be considered, with the cosmic dose rate being about 300 micrograys per year at sea level, and changing depending on altitude and the thickness of the covering sediment. It does get a bit difficult to understand once the quantum physics get involved, but essentially the trapped electrons in the sample are measured when the sample is subjected to an external magnetic field and dosed with specific wavelengths of microwave radiation. That changes the spin rotation of the electrons to either match the surrounding magnetic field or be opposite of the field. By measuring the changes in the magnetic field, scientists are able to determine the dose of radiation that the sample was subjected to since being formed or reset. Together, electron spin resonance, thermoluminescence, and optically stimulated luminescence dating are known as trapped charge dating methods. Thermoluminescence dating essentially works the same way as ESR dating, but the trapped electrons are measured by heating the object to free the trapped electrons, which results in the emission of light. The amount and type of light produced can be measured and translates into accumulated dose of radiation for the sample. Similarly, optically stimulated luminescence will use exposure to blue-green light to stimulate the release of the trapped electrons, which then give off photons which can be detected. Trapped charge dating methods essentially determine the last time the sample was exposed to sunlight, as direct exposure to the sun will reset the clock. Since we are dating the last exposure to sunlight, this can be used to directly date sedimentary rock, though certain limitations of this method make it useful only to about 1 million years. These methods are typically applied in archaeology and paleontology and can be used to directly date some of the artifacts that we have, like pottery. The process of firing ceramic pottery serves as a reset for the electrons, so these methods are great at getting us ages for these types of artifacts. Next up, we can finally talk about radiometric dating, the one that everybody was probably itching to get to in the first place. So, some atoms that are fairly ubiquitous throughout the Earth in small quantities go through a process called radioactive decay. Radioactive decay is when the nucleus of an unstable atom, the nucleus being the part that contains the protons and neutrons, spontaneously breaks down. This happens because these atoms do not have enough binding energy to hold the nucleus together. There are 3 types of radioactive decay: alpha, beta, and gamma. In alpha decay, the nucleus loses 2 protons and 2 neutrons, which are known as an alpha particle. If you'll remember back to high school chemistry, the number of protons in a nucleus are what determines what element it is, so the loss of 2 protons bumps it down the periodic table. So for instance, uranium-238, when it sheds an alpha particle, becomes thorium-234. Beta decay has 3 main subcategories, but here I'm going to be sticking to beta minus decay, as that's the one that everybody is probably a little bit more familiar with, since that's what takes place when carbon-14 decays. Beta minus decay is when a neutron sheds an electron and becomes a proton, which then causes the element to move up the periodic table. For instance, when carbon-14 sheds an electron and converts a neutron to a proton to become nitrogen-14. Gamma decay is a process that usually follows alpha and beta decay, but it doesn't result in transmutation or the changing of one element into another through the change in the nucleus, because gamma decay only emits energy and not matter, so I'm going to be pretty much leaving gamma decay alone for this video. And just as a refresher, the different numbers by the element names, like the 14 and carbon-14, represent which isotope of that element we are talking about. What element an atom is determined by how many protons are in the nucleus, but there can be different numbers of neutrons for the same element. The atomic number of the element represents the number of protons, so carbon has an atomic number of 6. So any atom with 6 protons will be a carbon atom, but it can have anywhere from 2 to 16 neutrons. So carbon-12 has 6 neutrons and 6 protons, while carbon-14 has 8 neutrons and 6 protons. So when the parent isotope carbon-14 decays, 1 neutron becomes a proton, meaning that when you add up the neutrons and protons, you still have 14 total, but now you have 7 protons and 7 neutrons instead of 6 and 8, giving us nitrogen-14. All else being equal, atoms tend to be more stable when the number of protons equals the number of neutrons. So typically, the greater the difference in the number of neutrons when compared to protons in an atom, the less stable that atom will be. The half-life of a particular element is how long it takes for half of the original concentration to decay. So if you have 1,000 atoms of carbon-14, which has a half-life of 5,730 years, then after the first half-life has passed, you will have 500 carbon-14 and 500 nitrogen-14 atoms. After another 5,730 years, you will have 250 carbon-14 and 750 nitrogen-14, and so on. The half-lives of the various unstable isotopes are, as far as geology is concerned, constants. It is possible to change the rate of decay, but it requires such extreme manipulation as to be completely unreasonable to think that that sort of change could have happened naturally. For instance, if you accelerate the isotopes to near the speed of light, time dilation will slow down to the decay rate as we perceive it. But last time I checked, rocks in the ground don't move at significant fractions of the speed of light relative to us while sitting in nature. Some forms of beta decay, particularly electron capture, where a neutron absorbs an electron to become a proton, can be altered by changing the state of its surrounding electrons, but the change in half-life due to changing electron states is typically significantly less than 1%. Hypothetically, you could strip away all of the electrons from such an element and keep it isolated from electrons, causing its decay rate to become infinite, but such an electrically insulating environment isn't a very plausible occurrence. And even though we are pretty certain at this point that radioactive decay rates are, for our purposes, constants, it is not something that is just blindly accepted. It is constantly being challenged and verified. For instance, in 2010, physicists at Stanford and Purdue universities noticed the differences in measured decay rates over several experiments, and proposed that this could be due to a change in the decay rate rather than measurement error. But further analysis has determined that the instrumental instabilities due to environmental conditions in the laboratory were the cause of the differences in measurements. That is, the instruments that the scientists were using to measure the decay rate were affected by environmental factors like ambient temperature and humidity. So to date something radiometrically, we take account of how many of which atoms are contained in a rock. So for instance, if we're using rubidium-strontium dating, we will count up how many rubidium-87 atoms there are, and how many strontium-87s there are, and use some relatively simple math to figure out how long it would have taken for that much strontium-87 to form from an initial amount of rubidium-87 that is calculated based on how much strontium is present. And we can be reasonably certain that no new parent or daughter isotopes were introduced into the system since the rock formed, because the conditions that would allow for that kind of contamination would leave signs of metamorphism on igneous rock, and of course we also cross-check the results with other dating methods like relative dating based on stratigraphic relationships to see if they make sense. But let's ignore that for a moment. Let's just say we have no other way of confirming the ages of the rocks, and we can't rule out contamination just based on the parent and daughter isotopes that we find. That's where isochron dating comes in. Isochron dating is useful when using elements that decay into a daughter element that has a second stable isotope, like strontium. In rubidium-strontium dating, rubidium-87 decays into strontium-87, and in the decay process we know it will always decay into strontium-87. But of the 4 isotopes of strontium, 2 are stable, strontium-87 and strontium-86. In fact, strontium-86 is completely non-radiogenic, that is, it is not the product of any decay chain. So in isochron dating, measurements are taken from several different samples which are all formed at the same time from a common pool of materials. Rocks that contain several different types of minerals are really good candidates for this. They then calculate the ratio of the parent isotope to the non-radiogenic isotope of the daughter element, and plot that on the x-axis of a graph. The y-axis is the ratio of the radiogenic daughter element to the non-radiogenic. When the ratios from these samples are plotted on the same graph, there should be a linear relationship with a positive slope. Isochron dating works because in a molten material, the rock crystals do not distinguish between the different isotopes of the same element when forming, so strontium-86 and strontium-87 will be included in the mineral formation at the same ratio. But the different minerals that formed at the same time will have different overall amounts of the two elements. So for instance, one mineral might form with 120 units of rubidium and 60 units of strontium, with there being 20 strontium-87 and 40 strontium-86. A second mineral forming in the same event will have different amounts of these elements, so let's say 60 units of rubidium and 90 units of strontium. But because the forming minerals don't differentiate between the isotopes of strontium, the ratio of strontium-87 to strontium-86 will be the same as in the first mineral. So of those 90 units, 60 will be strontium-86 and 30 will be strontium-87. And a third mineral might form with only 20 units of rubidium, but be strontium-rich, with 50 strontium-87 and 100 strontium-86. So the ratios of rubidium to strontium-86 will be different in every mineral, 3 for A, 1 for B, and 0.2 for C. But the ratio of the two strontiums will be the same for every mineral, in this case 0.5, so when you plot these numbers on a graph, you just get a straight horizontal line. Now let's fast forward through one half-life of rubidium, which is about 49 billion years. So yeah, my graph here isn't realistic since we're, collecting rock samples that are older than the entire universe, but it's just for illustration purposes, so bear with me. After one half-life, the amount of rubidium will be cut in half, so A goes from 120 down to 60, B goes from 60 to 30, and C from 20 to 10. For each atom of rubidium that decays, you gain 1 atom of strontium, so our values for strontium-87 rose to 80, 60, and 60 for A, B, and C respectively. So now our ratios have changed because strontium-86 is stable, so its amount has remained constant. So this is what the graph looks like after one half-life has passed. It is perfectly linear with a slope of 1. The steeper the slope, the older the rock. But what happens if there's a contamination or a loss of some of the radiogenic isotopes? Well, let's fiddle with the numbers for a bit. If mineral B loses some rubidium at some point, its data point slides to the left. If it is contaminated with extra rubidium, its data point slides to the right. And if we add some strontium-87, it hops up. And if we take rubidium from B while adding rubidium to C, they crisscross. In fact, there are all kinds of ways you can play with the data to make the points cha-cha real smooth. So what this all comes down to is that isochron dating will indicate whether or not your sample has been a closed system for its entire lifespan. Any contamination event or loss of either parent or daughter isotopes will cause the slope of the line to not be linear, and the chances of several different minerals being contaminated or losing their isotopes in identical amounts is extremely small, and the more samples from an event that you test, the less likely identical contamination becomes. This can actually also help us determine if the minerals we're testing were indeed formed in the same event. A separate event will have a separate profile for these isotopes, and so will mess up the graph if one of the samples is from a different event. And as a side note, if the decay rate of an element were to change at some point, that would also mess the graph up. Lastly, I'd like to touch on relative dating. Relative dating in geology relies on 6 principles of stratigraphy. The principle of superposition simply states that in an otherwise undisturbed sequence of sedimentary strata, the layers on the bottom are the oldest, and the layers above are younger. The principle of original horizontality states that rock layers that are deposited from above are laid down horizontally or mostly horizontally. It is possible that at the margins of basins, strata can slope slightly downward into the basin. The principle of lateral continuity is that within a depositional basin, the strata are continuous in all directions until they thin out at the edge of the basin. This is the one that lets us know that strata that are divided by a canyon remains continuous on either side of the canyon. The principle of cross-cutting relationship states that the deformation events like folds, faults, and intrusions that cut across rocks are younger than the rocks that they cut across. It's hard to cut across something that isn't there yet. So for instance, in this picture, the black rock is the youngest, the white rock is the middle child, and the gray rock is the oldest. The principle of inclusions is related, stating that when one rock formation contains pieces of another rock, the included rock is older than the host rock. Again, you can't include rock that doesn't exist yet. And finally, the principle of fossil succession is that groups of fossils contained in the strata are unique to the time in which they lived, so they can be used to correlate rocks of the same age across a wide geographic distribution. Setting aside the fossils for now, this basically means that we can look at a series of strata and figure out what order they formed in. So, as an example, this is a cross-section of the Grand Canyon. Using nothing but relative dating methods, we won't be able to determine the actual ages for the rocks, but we can at least figure out what order the rocks were formed in. Geologists can then determine how old the layers are in relation to each other based on their knowledge of how different types of rocks form. In fact, using relative dating methods were all that geologists had before the development of radiometric dating in the 20th century. Using relative dating methods, geologists in the late 1700s were able to figure out that the Earth is at least several million years old. And as our methods became more refined, and with the introduction of several methods of directly dating samples, we have narrowed down the age of the Earth to roughly 4.5 billion years. But it remains a fact that older sedimentary rock cannot be directly dated, so we must use the principles of stratigraphy to figure out the dates relative to rocks that can be directly dated. So we find places where the sedimentary rock is bracketed by igneous rock. That is, there's an igneous layer that is older than the sedimentary layers and one that is younger. They then use radiometric dating or isochron dating to directly determine the absolute age of the bracketing layers, and then use stratigraphic relationships to determine the ages of the rocks in between the brackets. And that brings us to index fossils. Fossils are always found in sedimentary rock because the conditions that result in the formation of igneous and metamorphic rock will always destroy anything that might leave a fossil. As such, fossils themselves cannot be directly dated. So in order to date the fossils, scientists will use the bracketing method with relative dating to figure out how old the rock that the fossils are buried in is. Since the burial has to happen relatively quickly in order for a fossil to form, it is a safe assumption that the fossils are the same age as the rock in which they are found. As paleontology progressed and as more and more fossils were discovered, it was found that certain organisms were very widespread over a large geographic area, sometimes worldwide. Using several dating methods, we have been able to determine the span of time in which these widespread organisms lived. These are known as index fossils. They are fossils that have been dated to a specific time period using absolute dating on bracketing igneous rocks, combined with relative dating to figure out when the sediment that buried them was deposited. Since these organisms lived in very specific times, they become useful for indicating the time period in which other unknown fossils are found, or for coordinating stratigraphic columns in separate locations, or for dating specific fossils that are known to span long segments of time. And I think I'll leave it there for today. In summary, we've learned that there are several different methods that can be used to date specimens, each of which have their strengths and weaknesses, but we have refined and continue to refine these methods to decrease the margin of error. In fact, uranium-lead dating is one of the most reliable methods and can often have a margin of error of less than 0.1%, which means that when we date a zircon crystal to 4.5 billion years with this method, we could be off by about a million years or so. So instead of being 4.543 billion years old, it might be 4.542 or 4.544 billion years old. So at the end of the day, we know that the Earth is old, and we are able to accurately determine the ages of fossils using several different methods. If you're enjoying this series, subscribe to be notified when new videos come out. You can also consider becoming a patron at patreon.com/ViceRhino. My patrons are what make this series possible. You can also follow me on Twitter and Facebook. See you next time. Hello and welcome to my channel. ViceRhino here. In today's installment of the Evidence for Evolution series, we are looking at the fossil record. We've been leading up to this for 2 weeks, but now it is finally time to dig in.
Come on, Josh!
So let's go! First, I would like to start by explaining how we determine the age of a fossil. I laid the groundwork for this over the last 2 videos, the ones on geology and dating methods, so if you haven't seen those 2 yet, I'd recommend giving them a look before continuing on with this one. So from those 2 videos, we learned how scientists determine the ages of certain types of rocks, but the rocks that can be directly radiometrically dated do not usually contain fossils, and just a minor correction here, I said metamorphic and igneous rock would never contain fossils in the last video, but you can find fossils occasionally in metamorphic rocks such as marble. But the process of metamorphism is not very kind to the fossils, so they are exceedingly rare. As for igneous, again, it is possible, but it's incredibly rare. And this is counting volcanic ash as sedimentary rock. It is technically igneous. One of the common phrases I heard was that it's igneous when it goes up and it's sediment when it comes down, because it behaves as a sediment when it comes down. Though, unlike other sedimentary rock, it can be directly dated. And on top of that, I completely missed pointing out in my last couple videos that volcanic ash makes really good stratigraphic markers. That is, they can often be fairly widespread because of how far the ash can travel from the source of the eruption, and because each eruption has its own unique chemical signature. So let's start here. If you find a fossil in a rock that was formed from volcanic ash, finding the date of the ash layer will give you the date of the fossil. Those are the easy ones. But what about fossils buried in, say, sandstone? Well, using radiometric dating techniques on sandstone will give you the ages of the grains that make up the sandstone rather than of the sandstone itself. But a sandstone cannot be older than its youngest zircon, zircons being crystals that are common throughout most types of rock. So that can give you a rough idea of how old the sandstone is, but it won't be exact. This is where we combine the principles of stratigraphy with different absolute dating methods to determine age ranges for each layer of rock, and the fossils that are found in the rocks were most likely buried by whatever depositional event ended up forming the rocks, so they will be the same age as the rocks in which they are found. That's all well and good, so now we have ways of figuring out when the different organisms lived and what environments they might have lived in, and even in some cases how they probably died. But what has that got to do with evolution? Well, the fossil record goes back billions of years into the past, and it gives us momentary glimpses into what life existed in the past. So if evolution is true, we would expect the fossil record to show us the actual evolutionary history of different organisms, and we would expect to see a trend of less diversity the further back in time you go. So let's just start by going as far back in time as we can and see what we find there. Hunting for old zircons can get us back to about 4.4 billion years, but there's no sign of any life that far back. The first signs of life showed up about 3.5 billion years ago in the early Archean, but fossil evidence of this life is quite rare until the late Archean with the appearance of stromatolites, which are rocks that are formed by the growth of several layers of cyanobacterial mats. The diversity of these organisms gradually increased throughout the Archean, peaking at about 1 billion years ago, and then starting to fade in both abundance and diversity some 700 million years ago, possibly due to the rise in herbivorous eukaryotes that would feed on them. This represents the majority of the fossil record of the Precambrian. It's almost exclusively single-celled organisms, with the first evidence of multicellular organisms appearing 2.1 billion years ago. So that's 1.4 billion years after single-celled life emerged. For comparison, Animals only emerged from the sea about 530 million years ago, so for the vast majority of the history of the Earth, there was nothing but single-celled organisms. And then for a good chunk of the rest of the history of the Earth, the most complex organisms were multicellular algae. And this brings us to the Cambrian period. The beginning of the Cambrian shows a massive diversification of life. This is when we see the appearance of almost every major animal phyla. That is, if we look at Linnaean taxonomy, it's the highest level before you get to the kingdom level, with kingdom being the division between animals, plants, fungi, protists, and Monera. So most major animal phyla make their first appearance here, but they would have looked very different from the members of those phyla that we see today. So for instance, this weird fishy eel-looking thing is known as Pikaia and was one of the earliest known chordates. So this guy represents you in that portion of the fossil record, and everything else with the spinal cord for that matter. And as a side note, this dude has only ever been found in Canada, so as it turns out, all chordates are Canadians. Yes, I know the landmass that we call Canada today didn't even exist back then, just let me have this. Back on topic, the Cambrian is when we see the first appearance of animals with mineralized skeletons and shells, though these still represent a minority of the fossils that we have from this time period. This is when we start to see the rise of different ecosystems, as these organisms' interactions were becoming more complicated. Predation caused the cyanobacterial mats that had been fairly ubiquitous up to this point to only survive in environments that their predators had not yet moved into. Tube-dwelling organisms evolved, which started to mix up the sediment at the bottom of the seafloor, causing what is known as the Cambrian substrate revolution, and essentially creating new ecological niches to be filled. Before the Cambrian, trace fossils left behind by early multicellular animals were very simple, just being horizontal lines along the surface. In the Cambrian, they started showing signs of more complicated movement, such as burrowing vertically through the sediment and just generally showing more variability in their behavior. So throughout the Cambrian, we see a relatively sudden increase in the diversity of life, and sudden here is a period of about 25 million years, so that's still an unimaginably vast amount of time, but it represents a measly 0.6% of the time that the Earth had existed up until this point. So geologically speaking, that is incredibly quickly. Now, why it happened so fast is a matter of some debate, but it is likely due to a combination of factors ranging from the increase in selection pressures caused by the development of predation, to the availability of oxygen for metabolic processes, to the first appearance of some of the important body plan-related developmental genes, to environmental factors. But while the Cambrian explosion gets all the attention for being the first rapid biodiversification event, it was tiny in comparison to the Great Ordovician biodiversification event. This happened after the Cambrian, and is when we first see the appearance of coral reefs and their accompanying ecosystems, though coral ecosystems would continue to be dominated by sponges and algae for quite some time after this. There was an approximately 300% increase in biodiversity during this time period. The Cambrian was amazing because it saw the emergence and experimentation with the various body plans. The Ordovician is where those body plans started to specialize. Trilobites, which first appeared in the Cambrian, continued to diverge in the Ordovician and increase in size. The largest trilobite found to date was 72 centimeters long. An increase in the diversity and types of plankton triggered a corresponding rise in diversity among suspension feeders. Both plankton and suspension feeders are important parts of the food web, so their diversification triggered still more diversification of the other organisms that were part of the same food web. Essentially, the diversification of the plankton and suspension feeders made nutrients more readily available for organisms that use them as food sources. The Ordovician was marked by shallow seas covering much of the continent of Gondwana, which served as fertile breeding grounds for species like the trilobites. It's also where we find the first signs of life making its way out of the sea and onto land, with the appearance of terrestrial arthropods and plant microfossils. And while this biodiversification was happening, the planet was very volcanically active, which provides geologists and paleontologists with several layers of volcanic ash that allow for easy and extensive calibration of the fossil record from this time period. The end of the Ordovician period is marked by an extinction event that was probably caused by the beginning of an ice age. Ice ages tend to lower sea levels, and as quite a bit of the life that developed in the Ordovician were living in shallow seas around Gondwana, that was bad news for them. This marked the beginning of the Silurian period, which saw the development of the first intelligent life, bipedal reptiles that hid deep underground to avoid what they saw as a coming extinction event. Doctor Who's a documentary, right? Now, I could keep going like this through all the time periods in Earth's history, but I'll just show this graph, which demonstrates the point that I'm getting at. As evolution progresses, the amount of diversity of the organisms living on the Earth increases. There are occasional extinction events that can remove quite a bit of the diversity, but aside from those events, the trend is clear and exactly what we would expect in light of the theory of evolution. But it's not just the fact that we see an increase in biodiversity, it's also the way the biodiversity increases. Once a body plan shows up in the Cambrian, we don't see new body plans developed to fill the same niches, we see modifications of that original body plan. Because the way evolution works, if something, like a body plan, can do the job well enough to allow the organism to survive, then that body plan will survive. If a slight modification to that body plan works just a little bit better, then that modification will gradually permeate the population. This is because a mutation that makes a small change to an existing structure is much less likely to be detrimental than one that makes a large, sudden change. So once a structure develops to begin with, it is much easier for evolution to work on this existing structure than to eliminate it and make a new one from scratch. And this is how we end up with vestigial features. So, if all this is the case, then we would expect to see evidence in the fossil record of organisms that are partway in between two stages. The two stages we are examining don't really matter. Evolution has no goals after all, so really any organism can be considered to be partway between stages. But there are certain transitions that appeal to us and are easier to pick out, such as the transition from sea to land. Tiktaalik is an excellent example of this transition. It has scales and gills like a fish, and its fins have the same ray bones for paddling as most other fish do, but there are also other bones in the fins that are more sturdy, suggesting that Tiktaalik was able to prop itself up in shallow water with them. It also has a rib cage that is a lot more sturdy than most fish, it has shoulders, and it has a neck. Jawed fish have a bone in their jaws called the hyomandibula. This bone eventually shrank and developed into the middle ear bone called the stapes. Tiktaalik's hyomandibula is smaller than its ancestors, but not nearly as small as the stapes bones are today. Ancient fish skulls are also jointed, which helps the fish with feeding and breathing underwater. Tiktaalik's skull is not quite as fused as a tetrapod skull, but it is more rigid and stiff than its fish ancestors. And while Tiktaalik probably was not the first vertebrate to venture onto land, it is from 375 million years ago, and we have found probable vertebrate footprints on what would have been land in 400-million-year-old rock, it is still an excellent example of the transition from sea to land. But how do we know that Tiktaalik actually is the ancestor of all land vertebrates? Well, we don't. In fact, it is quite likely that it was not the ancestor of the vertebrates, but that it was closely related to an unknown species that was the actual ancestor. It is estimated that only 0.1% of all the species to ever have lived are actually represented in the fossil record, and then the chances of a human finding those fossils are smaller still. So keeping these two facts in mind, it is highly unlikely that any given fossil that we find is an actual representative of a lineage that survived all the way to today. This is why, when you look at representations of the Tree of Life, or phylogenies, they usually don't show direct descent, but descent from an unknown common ancestor. So Tiktaalik is probably not our ancestor, but it is representative of a group of similar organisms that would have existed around the same time, one of which would be our actual ancestor, and Tiktaalik would have had a common ancestor with that unknown organism. It is possible that Tiktaalik is our direct ancestor, but the odds are heavily stacked against it. And this is the case for all of the fossil series that we see. The Pachycetus was probably not the ancestor of modern whales, but would have had a close evolutionary relationship to the organism that whales actually did descend from. Now, with that in mind, let's take a look at Pachycetus and the evolution of cetaceans, with cetaceans being the order of aquatic mammals including whales, dolphins, and porpoises. Pachycetus was a mammal that looked almost nothing like a whale. Its ankle bones contained a pulley structure that is only found in even-toed ungulates, like giraffes, so this placed it clearly within the lineage of the even-toed ungulates. Except it also had an ear bone that contained two features that are entirely unique to cetaceans. The involucrum, essentially a thick wall of bone covering the ear, and the sigmoid process, which is a bone that encloses part of the middle ear. Cetaceans are the only animals known to have structures like this, so it made sense to consider Pachycetus to be part of the evolutionary history of modern cetaceans. And as we discovered new, more recent fossils, this positioning made even more sense. Ambulocetus was obviously closely related to Pachycetus, including the cetacean ear bones and the ungulate ankle bones, but it was more specialized to an aquatic lifestyle with a stronger tail and paddle-like feet. Using stable oxygen isotope analysis, scientists are able to determine whether an animal drank freshwater or saltwater during its lifetime. Pachycetus drank freshwater. Ambulocetus drank a mixture of both. And when we come just a little bit closer to modern times, we find other proto-whales like Proseuglodon or Koochicetus, whose name I'm probably mispronouncing, but I like to think that it got that name from being ticklish. Coochie coochie coo, it's Koochicetus the ticklish whale. Not only do we find even higher levels of the oxygen isotopes that indicate they drank mostly, if not exclusively, salt water, but we find that the nostrils are starting to move back toward the middle of the snout instead of being at the tip of the nose, all while the hind legs are becoming smaller and less useful for walking on land. And as you get closer to modern-day cetaceans, they look more and more like modern-day cetaceans, which still have the remnants of pelvises hovering about where they should be if a whale were to grow hind legs. So, to recap, we learned over the last 2 episodes how exactly scientists are able to put together pictures of ancient ecosystems through geology and figure out exactly how old these ecosystems are. And the fossil record shows that the farther back in time you go, the less diverse the ecosystems become. And remarkably, we even have several fossil series that are good representatives of certain types of transitions. I only touched on Tiktaalik and cetaceans here, but we also have excellent fossil series showing the development of flight, the evolution of modern horses, and even modern humans. If you're interested in seeing human development, I highly recommend the series Systematic Classification of Life by Aron Ra. He goes through the fossil record and finds the different representatives of our ancestry all the way back to the first single-celled organisms that appeared 3.5 billion years ago. If you're mostly just interested in the human part of the series, start on episode 37, which covers the extinction event that killed the dinosaurs and led to the rise of the primates. That's it for today. Thanks for watching. Special thanks as always to my patrons, without whom this series would not be possible. If you'd like to support the series, head over to patreon.com/ViceRhino to pledge as little as $1 a week, which will give you some nice perks like early access to my Friday videos, discounts at my merch store, and 3D printed goodies. You can also follow me on Twitter at ViceRhino, or find the link to my Facebook page in the description. And I also have a P.O. box if for whatever reason you want to send me stuff. See you next time! Hello and welcome to my channel, ViceRhino here. Today's line of evidence for evolution is phylogeny, which, simply put, is the grouping of organisms together by their relationship into a family tree, or web in the case of organisms capable of horizontal gene transfer. Technically, when your 4th grader makes a family tree for a school project, they are constructing a phylogeny. But how can the construction of a phylogeny be used as evidence for evolution when it relies on the assumption of relatedness? Well, let's find out. Most phylogenies are hypotheses. That is, they are the proposed explanation for a narrow set of phenomena. The phenomena in this case being the appearance of relatedness. The reason most phylogenies remain hypotheses rather than becoming fact is because evolution is a messy process. There are often more than one phylogeny that can explain the appearance of relatedness, and most phylogenies deal with common ancestors that would have gone extinct a long, long time ago, long enough that their genetic material would not be preserved. And without genetic testing to verify it, it is entirely possible that a structure that appears to be homologous could actually be analogous instead, being the result of convergent evolution. And in more distantly related organisms, even genetic testing is not perfect. So phylogenies must be analyzed with several processes, the most common of which is known as parsimony analysis. Parsimony is a principle that is similar to Occam's razor. When there are multiple explanations for a phenomena, the one that relies on the fewest added assumptions is preferred. So if you're looking at 4 taxa, that is, a taxonomic group of any rank, so it could be species, order, family, phyla, and the like. So if you have 4 taxa, A, B, C, and D, then with respect to taxon A, there are 3 potential phylogenies. That is, it could be most closely related to B, most closely related to C, or most closely related to D. Parsimony analysis would examine all 4 taxa and determine which structure of relatedness is most likely based on the different criteria depending on what data you're working with. For instance, genetic data will be looking at, you guessed it, DNA, whereas fossil data would be looking at morphology. So when looking at our 4 taxa, you could arrange them in 3 ways, in 3 phylogenies. To oversimplify it, the analysis looks at which of the phylogenies results in taxon A having the most in common with whichever taxon is closest to it in that phylogeny, and this one will be the one proposed as the most parsimonious. Sometimes this can be relatively easy with morphology. There has never been an instance of anything analogous to the spine convergently evolving, so we know that if an organism has a spine, it fits into the vertebrata clade. Sometimes, though, it can be a bit more tricky. The clade Uniconta consists of organisms whose flagellated cells have one singular flagellum. But it would be entirely reasonable to think that it's possible that an organism which has multiple flagella could evolve in such a way as to lose all but one flagellum, meaning that a biconta, an organism with two flagella, could potentially evolve into an organism that we would have no way of knowing just by looking at morphology does not belong in Uniconta. This is why we have to look at character analysis. The phylogeny that involves the fewest number of character changes is most likely to be the correct one, so complex and in-depth phylogenies will always be subject to revision as more discoveries are made, but one thing is certain. When you organize organisms based on shared character traits, a nested hierarchy structure emerges. Now, at this point, it's helpful to know the difference between a monophyletic and paraphyletic group. A monophyletic group is an exclusive group of organisms in the same taxon that all share a most recent common ancestor, and which includes all descendants of that common ancestor. A paraphyletic group describes a group of animals that all share a common ancestor, but which does not include all of that ancestor's descendants. So, for instance, chordate is monophyletic. All chordates share the same chordate ancestor, and no group has independently evolved a notochord, and no chordate has ever lost the notochord. So every descendant of the original chordate ancestor is included in this group. But reptilia is paraphyletic. Mammals, birds, and crocodiles all share a common ancestor, but the characteristics used to describe a reptile do not apply to birds or mammals, so they are paraphyletic. To refer to the group that includes reptiles, birds, and mammals, it would be more proper to use the monophyletic term amniote, as that characteristic encompasses all of its descendants. Usually this problem comes about as a clash between the scientific term for an organism and the common language term. For instance, the term wasp is used to refer to several different organisms throughout the Hymenoptera group, but excludes certain others like ants and bees. As such, the term wasp is not scientifically helpful. Same goes for fish. I have heard it said that because of cladistics, we are all still fish because you don't outgrow your ancestry. And while it is true that you don't outgrow your ancestry, that doesn't apply to the colloquial term like fish, because the definition of fish necessarily excludes some descendants of organisms that would be our common ancestor with fish. Now that's all well and good, but why is this evidence for evolution? Isn't it all based on the assumption of relatedness? Yes. And that's actually part of why it is evidence for evolution. Evolution predicts that different species would be related to each other. A consequence of this prediction is that related species would have characteristics in common, and the closer the relation, the more characteristics are in common. Cats and dogs have more in common with each other than they do with rabbits. Cats, dogs, and rabbits have more in common with each other than they do with armadillos. Cats, dogs, rabbits, and armadillos have more in common with each other than they do with birds. And so it goes. The farther back in time the common ancestor of two extant species, the less those two species will have in common. But if there is no relatedness between different species, if they do not share a common ancestor, then it is reasonable to expect that similarities will show up in populations with no discernible pattern. A cephalopod might have a spinal column, a turtle might have an amniotic egg, an insect might grow an internal skeleton, a mammal might grow feathers, a bird might grow fur, and so on. These characteristics are known as synapomorphies, and it's more than just superficial similarities. After all, a dolphin and a tuna fish share many characteristics superficially, but they are not very closely related. This takes us back to homology. Using the different methods that we have at our disposal, we can determine whether a character we are looking at is similar because of ancestry, or whether it developed all on its own. And when you arrange animals by their shared, derived characters that are determined to be homologous, it ends up looking remarkably like a phylogeny. The phylogenies usually have, at the base of each group, a character that is shared by all of the descendants of the organism that first developed that character, but none of its ancestors. Usually the exact organism is unknown, so the common ancestor will be represented with the name of the character rather than the name of a species. Once a phylogeny is constructed, it needs to be analyzed for accuracy. Because we can't always know for certain if a character is homologous or analogous, there will always be a degree of uncertainty when it comes to placement on the tree. For instance, if we weren't able to trace the embryological development of the wing, and if we didn't know about the underlying bone structure of different wings, then we would have no way of knowing if bat wings and bird wings were homologous or analogous. If we didn't know, then just the fact that they appear to be a shared character would place them close together on a phylogeny, though we do know that bats belong closer to the wingless mammals and birds belong closer to the wingless dinosaurs than to each other. That's where the different methods of assessing the accuracy of the tree come in. There are several different ways of checking phylogenies for accuracy. I touched on the parsimony method earlier. For our bat and bird example, we would examine bats, birds, and a third animal, let's just say another mammal for simplicity's sake. A tree that places bats close to birds would show one character in common, the wings. A tree that places bats with the other mammal shows many more characters in common that don't fit with birds. Fur, mammary glands, live birth, the structure of the ear, and placentas, just to name a few. So the tree that places bats with mammals is more parsimonious than the one that places them with birds. There are also maximum likelihood methods for developing phylogenies, which they will infer rates of evolution directly from the data and determine the tree that best describes the data given those inferred rates. So maximum likelihood actually develops a tree from raw data that has the highest probability of occurring. There are several other methods, and one important fact here is that, usually, different methods of producing phylogenies that rely on different underlying assumptions will end up agreeing on phylogenies that are highly similar when looked at statistically. But how do we know that these phylogenies are actually accurately representing real relationships? Well, because it's been tested. There have been several instances of researchers breeding mice, bacteriophages, viruses, and plants, keeping track of the actual phylogenetic relationships, and then using the various methods of reconstructing unknown phylogenetic trees, and have found them to be highly accurate. For instance, a bacteriophage was propagated and exposed to a mutagen, and each lineage was tracked. At the end, there were over 135,000 possible phylogenetic trees. They then used 5 different methods for inferring phylogeny. The person on their team who did the inferential phylogeny was kept in the dark as to what the actual recorded phylogeny was, in order to avoid skewing the results with bias. All 5 methods used produced an accurate phylogeny. And if all that isn't enough, phylogenetic analysis has met the 5 criteria for admissibility of expert testimony in the United States. Those criteria being that 1, it can and has been tested, 2, it has been subjected to peer review and publication, 3, it has a known error rate, 4, there currently exists actively maintained standards controlling its operation, and 5, it has the backing of the scientific consensus. There are several cases where phylogenetic analysis has been used as evidence that has resulted in convictions. So just for fun, let's go through a very much oversimplified phylogenetic lineage leading to modern humans. We have nuclei in our cells, so we are eukaryotes. Our cells with flagella have one flagellum, so we are uniconts. We are multicellular eukaryotes, so we are metazoans. Our bodies have bilateral symmetry, so we are bilaterians. Our embryos have notochords, so we are chordates. We have a sense of smell, so we are part of the olfactores. We have skulls, so are in craniata. We have spines, so are vertebrates. Skipping down past some of the more technical ones, we have jaws, and so are gnathostomes. We have bony rather than cartilaginous skeletons, so are osteichthyes. Skipping down past even more of the technical ones, we have 4 limbs, so are tetrapods. At this point, it's necessary to point out that I am getting these images from the Phylogeny Explorer project, which is an incredibly ambitious project that seeks to be a comprehensive map of all known life forms. It is incomplete in some places, and so that's why there are placeholder names for splits that we know happen but haven't necessarily named yet. So that's where unnamed tetrapod 1 comes in. Take out the unnamed splits, you'll still end up at humans. Our legs are not sprawled out sideways like crocodiles, so we are therapsids. Our teeth are differentiated, so we are cynodonts. We have hair and feed our babies with milk, so we are mammals. We have placentas, so we are placental mammals. Our hind legs are dominant, and we have opposable thumbs, so we are primates. The exterior of our noses are dry, so we are Haplorini. Our nostrils point downward, so we are Catarrhini. We are completely bipedal and use tools, so we are Homo. I have skipped over many divisions in this phylogeny, each with a set of defining characteristics, and for the ones I did show, I gave only brief descriptions of the defining characteristics, where there were often several more. If you want a more comprehensive tracing of our lineage and phylogenetic grouping, I highly recommend Aaron Ra's currently 48 video series called The Systematic Classification of Life. Even in 48 10-ish minute videos, his are still oversimplified. So we fit the descriptions of all of these categories perfectly, and our closest ancestors, chimps and bonobos, fit them all perfectly as well, at least until we get to the split that led to their clade Pan. Our next closest relatives, gorillas, fit the exact same series of characteristics right up until they diverge at Homininae, and so on. The farther back you go, the more relatives can be included in the phylogeny. So at the end of the day, Phylogenies in isolation are not evidence for evolution, but when you combine them with the other methods that we have of determining relationships, they lay out a remarkably accurate map of relationships among all life on Earth, and they have been regularly put to the test to ensure that they aren't just coincidentally matching up. The theory of evolution is the best explanation as to why life appears to be related. Thanks as always to my patrons who make this series possible. If you'd like to support the show, you can do so for as little as $1 per episode at patreon.com/ViceRhino. You can also follow me on Twitter at ViceRhino, and a link to my Facebook page as well as my P.O. Box address are in the description. See you next time. Hello and welcome to my channel. ViceRhino here. Today we're taking our first dive into genetic evidence for evolution and looking at sequence homology. This one will rely heavily on some of the concepts that I laid out in the videos on homology and phylogenetics, so if you haven't watched those two yet, I'd recommend starting there. Assuming you're all caught up, let's go! Simply put, sequence homology is the study of DNA, RNA, and protein sequences in relation to their shared ancestry. There are three basic ways that genes can diverge. There could have been a speciation event causing one copy of the gene to be subject to different selection pressures in different populations and end up with two or more similar genes that are known as orthologs, a duplication event whereby genetic material is mistakenly copied more than once and these two copies evolve in different ways, with these genes being known as paralogs, or through horizontal gene transfer, which is a subject that can make this far more complicated than it needs to be for now, so I'm going to largely ignore it. Suffice it to say that horizontal gene transfer is an important evolutionary mechanism, particularly among single-celled prokaryotes, but it makes tracing gene homology difficult in organisms that utilize it heavily. Sequence homology will look at genetic or protein sequences and compare them to similar sequences found in other organisms, and then using the methods I described in the phylogenetics video, they will build a phylogeny based around shared genetic material between organisms. But the genetic material will not be identical. Depending on how long ago two species diverged, there could be significant differences between homologous genes in different species. Of course, this is exactly how the phylogenies are built, by figuring out the evolutionary path that requires the least number of changes between steps, essentially. By comparing genomes between species that are very different from each other taxonomically, it has been found that orthologous relationships can be established for the majority of the genes of their genomes, which essentially demonstrates common ancestry. It was proposed in the 1960s using protein sequence analysis that there had been two whole genome duplication events in between the common ancestor for all vertebrates and the common ancestor for all tetrapods. Protein sequence analysis is an indirect method of determining gene sequences, as specific amino acids are produced by specific base pair sequences. The same amino acid can be produced by different base pair sequences, so it is not as exact, but it is an excellent tool for determining evolutionary relationships nonetheless. I'll get into that a little bit more a little later on. By comparing proteins in vertebrates that are important in the production of hemoglobin and measuring the amount of genetic material present in various species, three scientists, Ono, Wolf, and Atkin, were able to determine that the current vertebrate genome was a result of two whole genome duplication events, one before the divergence of the common ancestors with the jawless fish, and one after. This was done in 1967, before genome sequencing was really a thing, so they were relying mostly on protein analysis and comparison of amounts of genetic material. But further research has essentially confirmed this conclusion and discovered several other genome duplication events throughout evolutionary history. Because of how groundbreaking the original paper was, the divergence of genetic material as a result of whole genome duplication events came to be known as onologs, which are a subcategory of paralogs. Paralogous genes are quite possibly one of the strongest driving forces for evolution. It's an excellent way to derive new functions that are similar to already existing functions. You start with one gene that has a specific function, oftentimes a function that is critical to the organism's existence, so messing with it could be fatal, and then this gene goes through a duplication event, creating a second identical copy of the gene. Because the first copy handles a critical function, the second copy is free to mutate toward new functions. This is a great way for evolution to get a head start. Instead of building a new function from scratch, it can start with an already existing function and tweak it to its purposes. Of course, I am speaking here in terms of intent, but evolution really has no intent. But slow, random mutations are more likely to produce something new and functional if they start with something that is already functional. And the human genome is completely littered with the results of past duplication events. This chart is a mapping of duplication events in human chromosomes. All the blue lines are duplications that happened within a single chromosome, and the red bars correspond to duplications that copy genetic material in between the chromosomes. Purple bars are areas of the chromosomes that weren't even mapped out, and the gold bars are areas of unique genetic material that are surrounded by duplicated material that are more than 95% identical, greater than 10,000 base pairs in length, and separated by 50,000 to 10 million base pairs. These particular regions are known as rearrangement hotspots, and several of these regions have already been identified as relating to certain genetic disorders. This is one field where the study of evolution can potentially lead to medical advances. By analyzing our genome for this type of pattern, we can narrow down areas of the genome that are more likely to cause genetic diseases than other areas, potentially making the identification of problem areas faster and easier. Paralogs, while incredibly important to eukaryotic evolution, are actually quite difficult to use in the construction of phylogenies. One of the reasons for this is that genes don't necessarily evolve at the same rate as species, so if you construct an evolutionary tree for a specific gene, it will not necessarily match up with the evolutionary tree for the species in which you find the gene. Sometimes a gene can be conserved through a speciation event. For instance, both human beings and chimpanzees have a gene known as NANOG in the same location on chromosome 12. Obviously, humans and chimps are a different species, but the gene remains the same despite the several different speciation events that led to modern humans and to chimps. Not only do we have the same gene in the same location as the chimpanzees, but we have 11 broken copies of the gene scattered throughout our genome. Chimpanzees have 10, and of the 10 that chimps have, all of them are in the same places on the same chromosomes as 10 of the human broken NANOG genes. The 11th one, NANOG P8, is on chromosome 15 in humans, but is absent from chimps. This suggests that the 11 copies of NANOG in the chimp genome, 1 working and 10 broken, existed in the genomes of our common ancestors with chimps, while the 1 broken copy on chromosome 15 happened sometime after chimps and humans diverged. Back to the point, if you construct a phylogenetic tree using one single gene, it will not necessarily give you an accurate tree of speciation events, because there is no law of evolution that demands that all genes must change at the same rate as the species. Nevertheless, it is still possible to map out genetic relationships using paralogous genes. It's just not quite as straightforward as with other methods. Orthologs, on the other hand, are genes that evolve in different ways after a speciation event, which makes them easier to use when constructing phylogenies, as their genetic history will necessarily match up with those speciation events. Though paralogs can interfere here as well. Sometimes it can be difficult to distinguish between an ortholog and a paralog whose second copy has deteriorated beyond recognition, which is known as outparalogs. Tools do exist, however, that are designed specifically to filter out errors in the data that would be caused by both outparalogs and horizontal gene transfer. Tracing the history of orthologous genes is possible using several different methods, all of which essentially boil down to figuring out how much change has occurred in the gene that you're comparing since the speciation event that separated them. There are entire databases dedicated to storing information on orthologous genes throughout the animal kingdom for the purpose of reconstructing phylogenies that are as accurate as possible. They include information on thousands of organisms, over 100,000 orthologous groups, and almost 2 million phylogenetic trees. Now, the way we determine whether or not a gene is homologous is through statistical analysis. Because we now have several databases cataloging millions of gene and protein sequences, it's a fairly simple matter of plugging a new sequence into an algorithm and having it check for a match. In order to find a match, it has to be calculated that homology is more likely than the two sequences sharing similarities based on chance. And because of the sheer amount of data that we have in these databases, we have been able to determine that sequences that are not evolutionarily related have similarity scores that are indistinguishable from entirely random sequences. By analyzing the vast amount of data that is available, it has actually been determined that false negatives are much more common than false positives. That is, a sequence is more likely to be found to be unrelated to other sequences that it actually is related to, than it is to be declared related but actually not being related. One of the ways these missing homologs can be detected is to compare protein sequences rather than DNA sequences. It seems counterintuitive. After all, DNA is more exact, right? But it works exactly because protein sequences are less exact. Remember that which amino acid is placed on which location on a protein sequence is determined by codons, a series of 3 nucleic acids. There are several single nucleotides that can be replaced which will leave the protein unchanged. Because of this, proteins have a much slower rate of evolution than the actual genetic material itself, so when looking for homologs in distantly related organisms, the proteins themselves will have changed less than the underlying genetic code. It is possible to have two very different DNA sequences coding for very similar proteins, so if you do a direct comparison of the DNA, the algorithms might return a negative result because they kind of operate on a better safe than sorry, where I'd rather tell you they're not related when they are than the other way around. But if you compare the protein sequences, it's more likely to find the relationship. There are even different databases and algorithms that can be used to find homology within proteins. If two proteins are homologous, that does not mean they are identical. That just means they share a common ancestor. It is not only possible, but entirely likely that in homologous proteins, there will be internal parts of these proteins that are not homologous. So we have also developed algorithms to determine which sequences within proteins are homologous and which are not. Protein sequences that are homologous often have similar functions, but not always. Sequences that develop through orthology, differentiating after a speciation event without duplication, are more likely to share a function than sequences that develop through paralogy, the ones that duplicate before they differentiate. It is possible, using various statistical methods, to infer function from homology. This can be important for medical testing. Protein and gene sequences that can cause diseases in humans can be identified in various other species, and through experimentation on simpler organisms such as amoebas that have orthologs to our disease-causing proteins, new treatments can potentially be developed. So, in a nutshell, if we ignore horizontal gene transfer, genes can diverge in two basic ways. They can either be duplicated before a speciation event and then diverge from each other after a speciation event to create paralogs, or they can diverge as a result of the speciation event to create orthologs. We can use the data that we have collected about orthologous and paralogous genes to construct remarkably accurate phylogenies, and some of the information that we obtained through the study of orthologous and paralogous genes can be useful in understanding the causes of various genetic disorders, not to mention the potential development of treatments for these disorders. So now we have a field of study that not only provides evidence for evolution, but makes medical advances that wouldn't be possible if evolution didn't happen. So thanks for watching. Special thanks as always to my patrons who make this show possible. If you'd like to support the show, you can pledge as little as $1 per episode at patreon.com/ViceRhino, or find a PayPal link in the description for one-time donations. Links to social media and my website are in the description, as well as my P.O. Box address. See you next time! Hello and welcome to my channel, ViceRhino here. Today, we are going to be looking at evolution as it has been observed in laboratory conditions. There are several intriguing experiments to talk about, so let's just get right to it. First, let's just discuss how these experiments are set up. Studying evolution in the wild can be incredibly difficult because there are just too many factors that can't be controlled by the experimenters. It is possible to study the actual evolution, but to determine which selection pressures led to which traits is a lot more complicated, just because of the sheer number of selection pressures. So for today, I'm going to be sticking to lab settings. In a lab, the experimenters will have something specific in mind that they are seeking to test. As such, the experiments will be designed in such a way as to eliminate what are called confounding factors. Confounding factors are variables that could have an effect on the outcome of the experiment without necessarily having any indication for the experiment. So, to steal the example from Wikipedia, if you set up a study attempting to find out if the birth order of children is causally linked to Down syndrome, maternal age would be a confounding factor. Maternal age is causally linked to Down syndrome, and a third child is more likely to be born when the mother is older, so you might see a link between Down syndrome and birth order, but not because being born third can cause Down syndrome, rather it would be because of the confounding factor of maternal age. So when we are looking at experimental evolution, we are looking for experiments that have a good design to try and minimize these confounding factors. Keep in mind that the design of these experiments is not to force a preferred outcome, it is to test and see if the expected outcome matches up with reality. So when looking at one of my favorite experiments, where single-celled algae developed multicellularity as a heritable trait, the experiment was designed to figure out whether or not predation would eventually result in multicellularity. Since they were trying to figure out whether this specific mechanism is capable of causing multicellularity, the experiment was designed so that they were starting off with a wide range of genetic diversity, but that this diversity would be subject to as few selection pressures as possible in order to test the one selection pressure that they were interested in. So in this experiment, they took 5 independent colonies of algae and exposed them to a filter-feeding amoeba as a predator. They also had 2 colonies that were not exposed to the predator as a control group. The idea being that if there is a confounding factor that they have not accounted for, it should affect the controlled group as well. So if they see something remarkable in one of the experimental populations and in one of the control groups, then they know there's something other than the presence of the predator that caused that result. So the 5 colonies of algae are exposed to the predator, which, because of its feeding mechanism, puts a selection pressure on the algae to increase in size. 2 of the 5 colonies developed characteristics of multicellularity, strain B2 and B5. Notably, they achieve multicellularity through different methods. The B2 strain formed clusters of cells with no particular arrangement or number, while B5 preferred to form in clusters of 8 cells. The B5 population also went through different life stages, starting out as unicellular, going through a 4-celled stage, before finally ending up as an 8-celled organism. In both cases, the individual cells were grouped together within the extracellular matrix, which indicates that they formed that way through division of cells from within the cluster, rather than by being separate cells collected together to form an aggregate. Researchers then continued breeding the populations of B2 and B5 strains to ensure heritability, and they occasionally did separate them out into individual populations again. Now, the algae didn't just develop multicellularity, they developed new life cycles to accommodate multicellularity, and each colony went about it slightly differently. Wild-type and control group algae go through a stage where they lose their motility and then go through 2 to 5 rounds of mitosis, or cell division, before releasing single cells that are themselves motile and then returning to their normal single-cellular life. Now, of the strains that had developed multicellularity, 2 of them kept a life cycle that is similar to wild algae, but the remaining strains were distinct. In some of the strains, after a cluster of cells undergoes division, it stays close to its parent cells because it's embedded in the extracellular matrix, or ECM. They can sometimes capture additional cells and incorporate them into the matrix. In others, the cell clusters are made up of only direct descendants, meaning that they did not aggregate with free-swimming cells like some of the other strains did, but they stayed multicellular purely through reproduction. Because of this, in those strains, all of the genetic material within the clusters would be identical, meaning that these multicellular clusters would themselves work as units of selection, which basically means that they are a singular organism. There are no selective pressures that would favor one cell in the colony over another, as they share the same genetic material, so further selective pressures would be against the cluster of cells as a whole, compared to other similar clusters of cells. What's more, the process that these algae went through that led to the development of multicellularity as a stable, heritable trait matches the prediction that was made in another paper about how this might have happened in the wild. The algae from the experiment ended up looking morphologically very similar to the different species of algae that was discussed in the paper that made that prediction, as well as having similar methods of genetically controlling how many cells are in their colony. And as the icing on the cake, the trait of multicellularity took about 1 year to develop and has remained stably heritable for at least 4 years. This is fascinating evolutionary research, and it helps us discover the processes that could have led to the first appearance of multicellularity, and it demonstrates that it's not a huge, unobtainable step in the process, but is actually surprisingly simple. Next up, let's look at an experiment that has been going on for 32 years now, the long-term E. coli evolution experiment. E. coli are an excellent organism for experimental evolution, as they are easy to grow and count, they grow in simple environments that are easy to control, they have short generational times. I mean, this experiment is now past 70,000 generations since it began, and it's still going. They can be frozen, and they can survive the freezing and thawing process, so it's possible to have an ongoing archive that can be restarted at any time. So if you discover something significant that has changed in a population, you can go back into the freezer to find out exactly when the change started, and even restart an old population to see if the change will happen again. For this particular experiment, they used E. coli that had reproduced strictly clonally, meaning there is no horizontal gene transfer to muck things up, as it so often does. This also helps with avoiding cross-contamination. They can place a marker at a specific point on a bacterium's genome, and it will stay put within that population, so it gives an easy way to tell the different populations apart. And because it is so easy to work with, it has become one of the most well-studied species on the planet, so there is already a wealth of research and data available for it. In this particular experiment, they started with 12 populations of E. coli, all founded from the same ancestor so that they would be genetically identical, with the exception of the genetic genetic marker used to distinguish the populations from each other. And because of the presence of these markers, periodic freezing of colony samples, and the fact that they can revive from being frozen, it is very easy in this experiment for them to eliminate cross-contamination. If any is detected, they simply get rid of the contaminated sample and start over from a previous non-contaminated frozen generation. Because this experiment has been going on for so long, there have been dozens of publications and research papers that have come out of it that have helped us understand mutation rates, antibiotic resistance, the prevalence of beneficial mutations, the process of clonal interference, speciation, the development of new traits, and more. I want to focus here on the development of the ability to metabolize citrate. This is an important trait because one of the defining characteristics that makes E. coli what it is its inability to metabolize citrate in aerobic conditions. Checking for citrate metabolism is one of the main ways to test for the presence of E. coli. In the experiment, they grow the bacteria in a solution known as DM25, in which glucose is the only available food source that E. coli is able to utilize, but which does contain 250 milligrams of citrate. The E. coli is allowed to grow in the medium throughout the day, but runs out of glucose in about 8 hours, which causes them to stop multiplying. After 22 to 26 hours, 1 milliliter of the solution containing the E. coli is transferred into a new DM25 solution, and so it will resume growth. After 31,500 generations, some of the populations didn't stop growing after the initial 8 hours of glucose. Their growth just slowed down a bit. These populations have been tested on various other growth mediums, several of them with citrate as the only available food source, and these populations, known as CIT+, were able to survive on citrate alone. And because of the procedures used in this experiment to eliminate contamination, they were able to completely rule out the possibility of contamination in this case. And because they freeze generational samples every 500 generations, they were able to determine by rerunning the experiment with previous generations that the underlying mutation that ended up leading to the development of citrate metabolism happened around generation 20,000. This means that the ability to metabolize citrate was dependent upon a separate mutation that may have had a completely different function sometime just before the 20,000th generation. This is an excellent demonstration of the idea that the order in which an essentially random beneficial mutation arises can have a profound impact on the future evolution of that species. And as the original strain of E. coli was strictly asexual, all of the changes between the recent generations and the original strain are purely the result of mutation and genetic drift. Even ignoring the fact that one strain developed the completely new characteristic of being able to metabolize citrate, there have been numerous other examples of phenotypic and genetic evolution throughout the experiment. Because the main selection pressure in the experiment is the availability of glucose, all 12 populations developed mechanisms to improve their growth rates on the limited supply of glucose. They also decreased their lag phase when transferred into a fresh medium, they developed larger average cell sizes, and reduced their peak population densities. These are examples of convergent evolution, where similar means of adapting to a similar situation developed completely independently. An important thing to keep in mind here is that, at its core, this is evolution in action. I have seen people respond to the study with the criticism that the new E. coli are still just bacteria, but it is important to realize that bacteria are the most diverse organisms on the planet. It's hard to tell, but it's estimated that there could be upwards of 1 trillion species of bacteria on Earth. 500 to 1,000 species of bacteria live inside each human being. So, to dismiss a speciation event in bacteria because it's still bacteria is akin to saying that the differences between humans and elephants are irrelevant because they are both eukaryotes. Next up, let's take a look at a fruit fly experiment that's been going on for about twice as long as the E. coli experiment, but it hasn't shown quite the same dramatic results. Probably has something to do with the fact that while fruit flies reproduce very quickly, their generational time is nowhere near as fast as E. coli, so in 60 years they have had about 1,500 generations, rather than the over 70,000 generations that the E. coli guys are up to now. But fruit flies are an excellent way to study evolution in macroorganisms, as they do have remarkably fast generation times for a macroorganism. The fruit fly experiment began in 1954 and reared the flies completely normally with the exception of being in complete darkness. The fruit flies still look like normal flies, but their head bristles, which are used as sensory organs, are longer, they have a better sense of smell, and they're more adept at finding a mate in the dark when compared to normal flies. Despite these differences, they still maintain their circadian rhythm and are still attracted to lights. Perhaps the biggest problem with the dark fly experiment to date is that the control group of flies was wiped out by a faulty incubator, so there's no group of flies raised in identical conditions but exposed to light for the entire 60 years. So even though researchers have identified more than 220,000 single-point mutations and over 4,000 insertion or deletion mutations in the darkfly genome, it is difficult to determine which of these mutations are specific adaptations for adjusting to life in the dark. To get around this problem, researchers designed an experiment that would effectively reselect the mutations that were specifically adapted for life in the dark. They mixed populations of the darkfly with wildfly populations and kept one group in constant darkness and another group in normal day-night cycles. The idea here is that the recombination during sexual reproduction would cause the mutations whose occurrence had nothing to do with being kept in the dark to gradually become less prevalent in the population that was exposed to day-night cycles, but would be selected for in the dark population, so the genes whose prevalence was maintained through another 49 generations would be likely candidates for having an effect on the fly's success in living in the dark. Several of these candidate genes were found in several spots on the genome. So in these 3 experiments, we witnessed several key aspects of evolution in action. The development of multicellularity as a defensive mechanism, the ability to adapt to a new food source when the primary food source becomes scarce, and the selection of mutations that are favorable in an environment. All of these operate on the same basic mechanisms: random mutations, genetic drift, and natural selection. And these experiments have demonstrated evolution's ability to produce new features and functions, all while building on already existing features and functions. That's it for today. If you'd like to support this series, you can do so at patreon.com/ViceRhino for as little as $1 per episode. You can also find me on Twitter at ViceRhino and find links to my website and Facebook page as well as my P.O. Box address in the description. See you next time. Hello and welcome to my channel. ViceRhino here. Today we're covering a topic that dabbles in several other areas: biogeography. Biogeography is where we combine geology, geography, paleontology, and biology in order to figure out the history of the Earth as a whole. And that's just the area of biogeography that looks into the past. There are other aspects that are concerned with the present distribution of species and how they interact with their ecosystems, and how different factors within their ecosystems can exert evolutionary pressure. But we're mostly going to concern ourselves with the past here, so let's go! Put simply, biogeography is exactly what it sounds like. It is all about the study of the interplay between geography and biology. So the observation that Darwin made during his trip on the Beagle, that species on island chains were more closely related to each other than to their closest relatives on the mainland, was a form of biogeography. Islands are great for studying biogeography because they are capable of providing the reproductive isolation and strong selection pressures that are needed to allow species to diversify more rapidly. In the absence of large predators, small animals tend to grow larger, giving us, for instance, the giant tortoises and iguanas in the Galapagos. In the absence of vast grasslands as an abundant food source, large mammals tend to get smaller, as can be seen in the fossils of dwarf hippos, elephants, and deer that once lived on islands in Indonesia, the Mediterranean, and the Pacific. Geographic features are actually one of the main driving forces of evolution. If one population is constantly breeding, mixing, and mingling their genes with each other, then any novel mutations will either work their way through the entire population or be drowned out by the other genetic material. Evolution can certainly happen this way, but any selection pressures will be acting on the population as a whole. Enter physical barriers. If the population finds themselves suddenly separated by, say, a river, then even if both populations still experience identical selection pressures on both sides of the river, the genes that end up being fixed in each population will be different, as they are no longer continually exchanging genetic material with their cross-river cousins. As more time passes, the two populations become more and more genetically distinct from each other, leading to 2 or more species where before there was only 1. Islands put that process on steroids. The climates are often significantly different than the climates on the mainland, so when a population finds itself isolated on an island, the evolutionary process gets kicked into overdrive. In fact, a lizard population that was moved to a new island in 1971 developed a brand new structure in just 36 years or 30 generations. They developed cecal valves in their digestive tracts that cause the food to move more slowly through the intestines, creating fermentation chambers that allow the lizards to maximize the nutrients they can absorb from their food. The original population of lizards were mainly insectivorous, but the island they were moved to had an abundant plant food source, giving the survival advantage to the lizards who were best able to digest plant matter. matter. There were a number of changes they went through that relate to their ability to utilize plants as a food source, but new cecal valves was probably the most surprising. Cecal valves are entirely absent in the original population and are a structure that occur in less than 1% of all known species of scaled lizard. The behavior of the lizards was affected as well. With the increase in stability of their main food source, their population density also increased. And since their food source is now one that doesn't have to be chased, they stopped defending their individual territories. They developed larger heads than the original population with a stronger bite force, which is a good adaptation for chewing through tough plant matter. And the fact that this is an adaptation specific to helping with their new food source is evidenced by the fact that the change was most drastic in females. Females previously had smaller heads, and so they started out at a disadvantage as far as the new food source is concerned, so there was more pressure on them to develop larger heads than there was on the males. The limb proportions have also changed, with the new population that relies on immobile plants having slower sprint speeds than their insect-hunting cousins. So we not only have a catalog of living species that show the effects of island life on their populations, and a fossil record demonstrating these same effects, we also have experimental observation of these effects taking place. We don't entirely understand the mechanisms yet, but our island evolution research is suggesting that rapid evolution, as was observed in these lizards, might be the norm rather than the exception. But that isn't the limit of biogeography. We can also use it in conjunction with geology to help us reconstruct the Earth's past. By studying the fossil record and mapping out the distribution of the various species found there, we find that the continents must have been together in the past in order for the locations of these species to make any sense. For instance, cycad plants are a classification of plant that has a wide distribution across tropical and temperate environments, both today and in the fossil record. They're actually more abundant in the fossils than they are today. They look kind of like a cross between a fern and a palm tree, but they are not really closely related to either. Now, there are some plants whose seeds spread far and wide through the use of wind and water, but cycads mostly just use gravity. Their seeds plunk heavily to the ground and roll a short distance away, and will sink to the bottom of any body of water they might find themselves in. Because of this, their wide distribution across the continents only really makes sense if the continents themselves were joined together in the past, creating areas of dry land where these plants could be free to plunk their heavy seeds wherever they please. Tracing fossil plants and animals and mapping their distributions shows us that the continents must have been together in the past, and combine this with detailed geographic and geologic studies of the ocean floor, we have been able to figure out not only the mechanism behind continental drift, which resulted in the development of the theory of plate tectonics, but we've also been able to reconstruct the positions of the continents and supercontinents going back for billions of years. Since physical separation plays such an important role in evolution, the idea that the continents were once physically joined together helps us explain several evolutionary anomalies. For instance, the distribution of marsupial mammals. Through fossil evidence, we have been able to determine with reasonable amounts of certainty that marsupials first evolved in what would eventually become Asia, they spread through Pangaea to areas that would become North and South America, and eventually made their way around to the area that we know today as Australia. As the continents separated, the marsupials that lived in Asia, Europe, and North America went extinct, leaving only the ones in South America and Australia. Interestingly, the study of marsupials has also given us some insight into the development of the eye. Marsupials diverged from the rest of the mammals before the development of the vasculated retina. Their eyes are more similar to the eyes of reptiles, with the exception of the North American possum. This possum is the only marsupial to have developed a vascular system in the retina, and while it superficially resembles the vascular system found in the retinas of the other mammals, on closer inspection it is quite different, and is not as efficient a system for providing nutrients. They have also independently developed a structure known as the retinal tapetum lucidum, which is a structure on the retina that reflects light back through the eye to give it better night vision. This is the same structure that is found in other mammals, such as cats, but it is not homologous. The one in the other mammals is not attached to the retina, but to the choroid. It's likely that these two developments are closely related. A vasculated retina can definitely help with the development of a structure on the retina that can't exist without vasculature after all, though the initial selection pressure that led to the development of the vasculated retina in the first place are still unknown. The study of marsupials has also given us insight into the evolution of pregnancy. As far as pregnancy is concerned, marsupials are more basal, in the sense that it's thought that the increase in the supply of nutrients by the mother, rather than by a yolk sac, was one of the first developments that led to mammalian pregnancy. But the longer an embryo remained inside a mother's womb, the more likely it is that her immune system would start treating the embryo as a parasite to be attacked. So basal mammalian pregnancy was fairly short. In marsupials, pregnancy remained short, and they developed pouches for carrying their young externally, where they would be safe from the immune system. The placental mammals, on the other hand, developed mutations that suppress the mother's immune system locally in the womb in order to allow for extended pregnancy. These immune-suppressing mutations were likely the result of transposons, segments of DNA that do nothing but attempt to copy themselves into other parts of the genome, essentially parasites at a genetic level. This is a super oversimplified description of the evolution of pregnancy, but it'll do for now. This video is supposed to be on biogeography after all. So, this is a bit of a shorter one, but in summary, biogeography is an excellent explanation for the patterns of fossils that we observe in the fossil record, and the study of biogeography has led to many advances in our understanding in more than just evolution. Plate tectonics were described as a result of a biogeographical study decades before they were verified geologically. Biogeography also helps us understand just how rapid evolution can be, with completely novel structures showing up in just 30 generations. Studying the distribution of various species helps us understand how different selection pressures can act on a similar organism, and how different organisms can develop similar adaptations when exposed to similar selection pressures. Thank you so much for watching, don't forget to subscribe, and special thanks as always to my patrons. If you're enjoying this series, consider becoming a patron for as little as $1 per episode at patreon.com/ViceRhino. Patrons also get early access to my Friday videos, as well as a few other perks. Links to my store and social media, as well as my P.O. Box address, are also in the description. See you next time! Hello, and welcome to my channel. ViceRhino here. Today I'm giving you the 11th installment of my Evidence for Evolution series, and while this series does not have to be watched in order, this episode will be building on some things that we learned in the sequence homology episode, so if you're new to the series, or if it's been a while since you watched the sequence homology one, consider watching that one first. It's not critical, but some of the stuff here will make a bit more sense if you have seen it first. This episode is going to be all about endogenous retroviruses, or ERVs for short. This is, in my opinion, probably the strongest line of evidence for evolution that exists. So let's take a look. Let's start with an explanation of what exactly an ERV even is. A virus, as you probably already know, is a type of not quite alive organism that is incapable of reproducing by itself. It must use another organism's cells as its machinery for producing more of itself. It will insert its DNA into the host cell, and the normal transcription process of the cell will then work on the viral DNA to produce more copies of the virus. A retrovirus, on the other hand, uses RNA as its genetic material. A DNA virus just has to plunk its DNA down into the cell, and the normal chemistry of the cell will go ahead and transcribe that DNA. But a retrovirus can't do that. Because it uses RNA as its genetic material rather than DNA, it must first find a way to turn that RNA into DNA for the host cell to transcribe. This is why it gets the prefix retro, because it has to reverse transcribe its RNA into DNA before anything can be done with it. Retro as a prefix essentially just means backward. The method that retroviruses have developed for accomplishing this task is to use an enzyme that is aptly named reverse transcriptase to transcribe the RNA in reverse back into DNA. And then it used another aptly named enzyme called integrase to integrate its genetic material directly into the host's DNA. Unlike a DNA virus whose presence will eventually kill the cell, there is not necessarily a significant detriment for a host cell that becomes infected, and occasionally the infection can even be beneficial. Every now and then, a retrovirus will infect the gametes of an organism, and so enter the germline. This is when a normal old retrovirus becomes endogenous. Endogenous in biology refers to something that originates inside of an organism. So a retrovirus that is not passed on genetically is an exogenous retrovirus. The organism must become infected with that virus externally, while one that is passed down genetically is endogenous. The organism will be born with it, so it comes from inside the organism. ERVs are easy to identify because they always have the same structure, and this structure matches modern retroviruses that still give us grief, like HIV. On either end of the retroviral DNA will be two identical sequences known as long terminal repeats, or LTRs. These sequences are essentially the first bits to infiltrate the host DNA, and their job, in an oversimplified nutshell, is to make space for the rest of the retroviral DNA in between them. In between the two LTR sequences, we find the genetic material to encode for three main proteins: group antigens, abbreviated GAG, which are the proteins that make up the core of the virus, POL, which codes for the reverse transcriptase, and ENV, or ENV. I'm not sure if I'm actually supposed to be reading these out or just saying the letters, but ENV is the envelope protein, which codes for the envelope that makes up the body of the virus. Each individual virus is different, but they all have these same structures. So, what do viruses hijacking our systems to reproduce have to do with evolution? Well, it's quite simple, really. ERVs are essentially inserted into the genome of the host species at random, with there being, conservatively, about 50 million potential insertion points for the average grade ape genome. With the size of the genomes and the random nature of the ERV insertion, the chance of two identical ERVs being inserted in the same location are minuscule. So by examining the various ERVs in the genomes of the different apes, we can begin to construct a phylogeny. I think an analogy would be helpful here, so I'm going to go ahead and steal one from Pelagia. It's the best analogy I've heard so far, and I don't really see a need to reinvent the wheel here, so here we go. So let's say you have a hypothetical collection of 10 books that are all different from each other but have differing degrees of similarity. In looking at the books, you can see that page 20 is identical across all 10 books. But on that page, 7 of the 10 books have a stain of pasta sauce that is identical in shape and location. While there is an incredibly minuscule chance that 7 individual pasta sauce splattering events created an identical splattering pattern on the page, it is far more likely that there was a splatter on the original and then the other copies were photocopied from that. So the 3 unsplattered copies were made, then the splatter happened, then the 7 splattered copies were made. So we can see that all 10 books shared a common book ancestor, but the 7 splattered books are more closely related to each other than to the unsplattered books. So then we notice that in the splattered books, page 24 is also identical, but in 4 of the books, there is an identical scribbling in the corner on page 24. Again, the most parsimonious explanation is that while these pages were being photocopied, 3 of the copies were made before the scribbling, and 4 were made after. And we can also determine that the scribbling happened after the pasta sauce splatter. But in the books that lack the scribble, page 17 is also identical, and 2 pages of page 17 have an identical ink stain on them. So using this information, we can arrange the books in a nested hierarchy, and we can determine the relationship each book has to the other books, and we can also construct a timeline of when these page-altering events would have happened, roughly. In this analogy, the text of the book represents the genome of the organism, and the external modifications to the book, the stains and scribbles and whatnot, are the viruses. Now, of course, this analogy is imperfect because books don't reproduce sexually last time I checked, so they're not actually reproductively related to each other, but it does kind of help get the idea across. With each ERV, there are 3 individual data points that can be used to construct an independent phylogeny. First, there is just the general distribution of ERVs as a whole among the different taxa, which provides the overall timeline. Second, using the methods that I described in the sequence homology video, we are able to construct phylogenies based on the differences between the same ERV in the same locations on the genomes in 2 different species. If you'll recall, after 2 populations are isolated from each other, their genetic material will accumulate different mutations. These differences can be statistically mapped out in the creation of a phylogeny, and the presence of an ERV makes this relatively easy. Third, each ERV has 2 LTR sequences on either end. Because of how retroviruses work, these LTR sequences need to have been identical when the virus was inserted, or the viral insertion would simply have failed. Because we know that each LTR had to be identical, we can use both LTRs to construct 2 independent phylogenies, and any variation in the phylogeny will necessarily be the result of an event other than the accumulation of neutral mutations that these phylogenies rely on, and so provides a powerful error-correcting mechanism. So because each ERV that is present across multiple species can be used to construct 4 independent phylogenies, one from the general distribution of the ERV, one from studying the sequence homology of the ERV, and 2 from the LTRs, we have 4 independent phylogenies that can be used to check the work of the other 3. And on top of that, there are literally hundreds of these ERVs that have been used to construct phylogenies within the great ape families, all of which are in high agreement with each other. Now, if you'll remember back to my sequence homology video, a phylogeny based on genetics alone will not necessarily match up with a phylogeny made with species names. This is because our relatively arbitrary labeling system does not always perfectly reflect nature, and indeed it likely never will, as nature doesn't need to conform to our labels for it. So genes are free to mutate and change within a single species. As such, phylogenies of ERVs are usually constructed based on the ERV itself, rather than just slapping some species names on them. This is because it is entirely possible for 2 independent ERVs to become fixed in a population of the organism at separate times, without there having been a speciation event in between. This is actually why, when constructing genetic phylogenies from any data, the researchers will usually use the name of the gene, or the ERV, with some sort of other indicator used on the chart to represent the different taxa. Now, ERVs are not only good evidence for evolution, there is also evidence that ERVs have helped drive evolution. The LTRs of these retroviruses contain different regulatory sequences that will affect how the neighboring genes are expressed. So if they land in the right spot on a genome, they could, in one fell swoop, have a dramatic effect on how the host genes are expressed, or even if they are expressed. Usually, this results in disease, like cancer, and possibly autoimmune diseases. But very occasionally, it can have a beneficial effect. For instance, the virus's need of evading their host's immune system has caused them to develop immunosuppressant characteristics in some cases. Placental mammals have co-opted this effect to help with the local suppression of the immune system during pregnancy, so that way the mother's body will not attack the embryo. And this has been accomplished in slightly different ways in different mammals by co-opting different ERVs in a fantastic demonstration of convergent evolution. So ERVs, by their nature, are unlikely to be inserted into the same place on 2 different genomes. However, an insertion in the germline will yield offspring with the same ERV in the same location as the parent. Therefore, when we see hundreds of ERVs that are identically placed in the genomes of different animals, the most parsimonious conclusion is that these animals shared a common ancestor whose germline was infected with the retrovirus. And when we construct phylogenies based on this data, we can construct up to 4 phylogenies for each individual ERV, and ERVs make up a significant amount of any mammalian genome, anywhere from 8 to 10%. We can then compare these hundreds to thousands of individually generated phylogenies and find that they are in high agreement with each other and high agreement with the phylogenies constructed with different methods. This indicates a relationship between the species that share this viral DNA over evolutionary time. In other words, common ancestry. Thanks for watching, subscribe if you're enjoying the series, and consider supporting the show for as little as $1 per week over at patreon.com/ViceRhino. See you next time! Hello, and welcome to my channel. ViceRhino here. Today we are going to continue on with my formerly regular but now occasional series on the evidence for evolution. This installment will be covering transitional species, but rather than hash out all the transitional fossil sequences that we've all heard a bunch of times, I thought I would take it in a different direction and point out some currently extant species that have traits that could place them in different categories, making them appear to span the gap between those categories. So let's go! Let's start off with an animal that, if you follow me on Twitter, you may have already heard me talking about. I might have gotten a little bit salty at the fact that this fish is an amazing example of a living transition, and has even been experimented on by scientists in order to learn about the evolutionary development of various tetrapod characteristics, but nobody seems to be talking about it. When bringing up living transitional organisms, the go-tos all seem to exclude these guys. So I had never heard of them when researching any of my videos. It wasn't until I started getting into fish keeping that they came up, and even then, it was just a cool monster-looking fish that can keep an arowana company without getting eaten. They didn't go into just how cool these guys are from a scientific perspective. I'm talking, of course, about the Bichir. And yes, I am probably saying that wrong. And yes, I have looked up how to pronounce it. The problem is that everyone tells you a different pronunciation is the proper pronunciation. These guys are known by my kids as the monster dinosaur puppyfish, or just as Falcor when we're talking about the Senegal Bichir.
So, little rhino, what's the next fish that we're going to get?
The monster dinosaur puppy fish named Falcor.
This is a very basal species of ray-finned fish, and are useful for studying early evolution of the tetrapods, as some of their key characteristics that make them a transitional species are likely derived from their common ancestor with the lobed-finned fish, meaning that their characteristics are quite similar to what these characteristics would have looked like in the lineage that led to the tetrapods. So what are these characteristics that make them so evolutionarily important? For one, they have lungs. And not the modified swim bladder style lungs that have developed in several other fish species, or the labyrinth organ that is a modified pharyngeal arch, but real, honest-to-goodness lungs. This would have been very helpful in their natural habitat, which is in swampy waters with minimal current. So in the wild, they live in water that is very poorly aerated, which makes sense. Breathing directly from the atmosphere would help them survive there. To breathe, they have spiracles on top of their heads, which they prefer to use to respirate at the surface, though when they don't feel safe, they can use their mouths for a quick gulp of air. There are many other aquatic species that have similar spiracles, including sharks, but they usually use them to push water past their gills while their mouths are otherwise occupied. This fact, combined with the bashir's tendency to breathe through its mouth when stressed, has led to debate about whether they even use their spiracles to breathe, but scientists have since shown that when the fish feels safe, it uses its spiracles for about 93% of its breaths, as opposed to 40% when it's stressed. Because their lungs are so similar to modern tetrapod lungs, in that they are similar in structure, they utilize recoil aspiration, and they're derived from a similar location on the embryo, they can provide us with a snapshot into the development of the first lung-type organs that would have eventually allowed tetrapods to move onto land. And this does answer the common question, what good is half a lung? Well, when it accounts for less than half of your respiratory needs, it turns out to be quite useful. And actually, it's worth noting that these fish, aquatic animals that usually live their whole lives in the water, will drown if they don't have access to air that they can breathe. But that's not where it ends with these guys. They have also been studied with regards to the development of tetrapod limbs. In particular, scientists were interested in studying phenotypic plasticity, which is the term used to describe the fact that some organisms will produce different phenotypes depending on their environment. I actually just had a discussion with this about my daughter the other day, as some water lettuce that we moved from my father's outdoor pond and put into our indoor aquariums has produced offspring whose leaves stay flat, as opposed to the normal upward curve shape of the parent plant. And while I don't know for certain if phenotypic plasticity is responsible for this, it served as the starting point for an educational discussion. Anywho, back to the monster dinosaur puppyfish, scientists hypothesized that phenotypic plasticity could have been responsible for some of the important changes that allowed stem tetrapods to move onto land, and they've been able to test this hypothesis through the bashir. Some species of bashir, notably the species that I am officially renaming the Falcor bashir, are able to survive and walk on land. So scientists raised a group of them on land and compared their morphology and behavior with their aquatic cousins. The way they move has changed quite a bit when compared to the aquatically raised fish. The land-raised fish held themselves differently and moved their bodies in a different manner, giving them a more efficient gait on land. These differences were hypothesized to be learned training advantages, where it's simply a matter of a fish being raised on land having more practice moving itself on land than a fish raised in the water. But there were anatomical changes as well. The clavicle and cleithrum, which are bones that connect the skull to the rest of the fish, were significantly different. The clavicles of the land-raised fish were narrower, with processes that were more pointed and 10% longer than the water-raised fish. The clavicle cross-section is thinner, and there was a reduction of size in the supracleithrum, which resulted in a weakened connection to the post-temporal bone and the cleithrum, which allowed for greater flexibility of the head independently from the rest of the body. In other words, the beginnings of a neck. Some of the changes between the aquatic and terrestrial bashirs mirror the development that was seen in the fossil record of stem tetrapods of the Devonian period, suggesting that these changes were not only important for the development of the tetrapods, but can also happen quite rapidly, with significant change happening here in just one generation. So from studying the bashir, we have found that phenotypic plasticity caused by environmental rather than genetic factors is a very important mechanism for large-scale evolution. It is important to note here that these changes were not genetic. They were likely related to different physical forces acting on their bodies, both from not being supported in the water and by using different muscle groups than their aquatic cousins. So we have phenotypic changes that are not the result of any changes to the genetics, so the physical features of the organism change in response response to the environment, regardless of the genetics, making them more suitable for land-based operations. This means that a sudden, drastic change in environment for an organism could very well be the starting point of that organism making the new environment its natural home, if it manages to survive the initial change. Next up is an animal whose omission from this list would get me piles of hate mail, the duck-billed platypus. These little weirdos are basically as close as you're ever going to get to the infamous crocoduck. They seem to be an animal that you would arrive at by hitting random on a character generator in a video game. And most of their features are primitive features that may have been shared by early mammals but later evolved into different features. They are one of only 2 species of mammals today that lay external eggs, the other being the echidna. They start out life in leathery eggs that are incubated by the mother. When hatching, they make their way out of the egg with the aid of an egg tooth, a very reptilian feature. Interestingly, they start out life with a set of teeth while they're nursing in the nest and not eating anything that requires teeth, and then they shed these teeth shortly after leaving the nest when they start hunting their own food, as their specially adapted bills have a flat surface of hardened gum tissue that it uses to crush the shells of its prey rather than chewing on them with teeth. Aquatic monotremes likely developed from terrestrial monotremes, and the first platypus-ish monotreme showed up in the fossil record about 110 million years ago, about 70 million years after the first mammals showed up. The platypus is one of the most basal extant mammals, having diverged after the development of lactation, but before the full development of internal pregnancy. Monotremes are also literally named based on the fact that they use one hole for defecation, urination, and reproduction, the cloaca. That is a very reptilian feature for a mammal to have. And yes, echidnas and platypuses, platypi, platypodes, whatever their plural is, they have penises, but their penises are used solely for semen, not for urination. And insemination of the females takes place through the cloaca. Also, echidnas have penises with foreheads, just FYI. Back to the platypus, genome analysis suggests that the monotremes diverged from the rest of the mammals about 166 million years ago, just 12 million years after the mammals evolved in the first place. While nursing, the baby platypus will suck directly on the abdominal skin of the mother as the females lack nipples, showing the evolutionary path that lactation took, with milk just being a secretion from the skin initially rather than passing through ducts and nipples. The protein composition during lactation changes the same way it does in marsupials, but not in most other eutherians, that is, the branch of mammals that are not monotremes. Given that marsupials are more basal than the rest of the mammals but less basal than the monotremes, it makes sense that these two groups would share some basal characteristics with each other. And the weird mashup between reptilian and mammalian characteristics in the monotremes can be observed on smaller scales as well. Their sperm are filiform like bird and reptile sperm, but the chromosomes in the sperm are arranged in a defined order like the rest of the mammals, but unlike birds. While the proteins that make up the sperm are mostly similar to those of other mammals, the epididymis, the tube in the body that transports the sperm, secretes a protein that is also secreted by the reptilian epididymis. Back to the chromosomes, if we examine the 52 chromosomes of the platypus, we find a mixture of reptilian and mammalian characteristics. The structure of the chromosomes is ranged in a very reptilian fashion, with just a couple of large chromosomes and lots of small chromosomes, which in reptiles are referred to as macro and micro chromosomes. Their sex chromosomes are somewhat homologous to the bird's Z chromosome system for reproduction. In birds, the female rather than the male has the shortened chromosome, and the egg rather than the sperm determines the sex of the offspring, though it is still unclear exactly how sex determination happens in the platypus. The platypus also has fewer than expected non-protein coding RNAs for a mammalian species, with their count being more similar to chickens than the other mammals. But when looking at coding DNA, it shares most of its commonalities with the other mammals. 82% of its genes were orthologs to genes in 5 other mammal species. But they also have conserved genes that have been retained in sauropsids, that is, reptiles and birds, and other non-amniote vertebrates, but lost in the eutherian and marsupial mammals. So all the mammals except for the monotremes. Its eggs are a weird mix of sauropsid and marsupial characteristics. The eggs are much smaller relative to body size than those of birds and reptiles. They hatch early, so the embryo does most of its growing outside of the egg, relying on lactation, as in marsupials. But the zona pellucida, the membrane surrounding the egg before sperm implantation, has 4 proteins that are identical to human zona pellucida, but 2 that are otherwise unique to birds, amphibians, and fish. The aspartyl protease nothespin is a gene that exists in fish and sauropsids, which is thought to be involved in processing egg yolk proteins, and is completely absent in the rest of mammals. And of course, I would be remiss if I failed to mention the venomous spurs of the platypus. There are a handful of other mammals with venom, but the platypus is the only mammal which does not deliver its venom with a bite. And here's where things get interesting. While platypus venom is very similar in composition to several reptilian venoms, it is the result of convergent evolution. They developed the venom all on their own. They did not inherit it from their reptilian ancestry. One of the common arguments leveled against evolution is the idea that scientists are just trying to make everything fit in evolution, so if they see something similar, they'll declare it to have an evolutionary relationship. Not so with platypus venom. Now, I could keep going with the transitional, not quite reptilian but not quite mammalian features on the platypus, like its immune system, genomic imprinting, repeat elements, the CpG fraction, the strangeness of its ability to electrically sense its underwater environment, and more, but suffice it to say for now that after not only a morphological analysis but also a genetic analysis, we find that the platypus, while definitely fitting into the mammal category, is much closer to the sauropsids than the rest of the mammals, showing several features that are shared with the sauropsids. As far as transitional species go, it could be said that every living species is transitional. It is a transition between what came before it and what their lineage will eventually evolve into should they survive. But usually the word transitional is meant in a way that would have a species representing two distinct groups of animals, and I chose two examples that I thought fit the bill pretty closely. We have a fish that can drown in water and be raised completely terrestrially, and we have a mammal that lays eggs and whose eggs aren't quite the same as sauropsid eggs, but also aren't quite the same as mammalian eggs, with characteristics of both, and a bunch of other characteristics that have them both straddling the lines between their various categories. I could keep going with animals that have partially developed features, like mudskippers, a species of blenny fish that live at least part of their life on land. Other blennies, which have been known to climb trees and can spend the majority of their life out of water. Labyrinth fish, which have a breathing organ that is modified from the last pharyngeal arch or gill slit. Lungfish, which, like the bashir, have pretty much fully developed lungs. Climbing perch, which can live for about 10 hours out of water. Walking catfish. Flying fish. Flying squirrels, flying snakes. There are so many examples to choose from for a living transitional form, and if you want to get into the nitty-gritty details, a case could be made for pretty much any living organism. The point is that one of the main arguments against the theory of evolution is a lack of transitional species, either in the fossil record or alive today, with objections being raised such as what good is half a lung or wing or eye? Well, we have living organisms today that essentially have half a lung, half a wing, and half an eye, so obviously they are at least a bit useful. Thanks for watching. Special thanks as always to my patrons, especially David Schinkel, Lynn Dobbs, Mark McManus, and What Jesus, who are the sturdy fins that allow the fish that is my channel to walk. If you'd like to keep me moving in an environment that I clearly wasn't made for, you can support the channel for as little as $1 per week over at patreon.com/ViceRhino. Other ways to support the channel, such as direct donation or my Amazon wishlist, are linked in the description, as well as my social media accounts and my P.O. Box address. See you next time!